Good morning, everyone. I just want to start by first welcoming all of you here to the Bach Institute. I know I've seen many old friends, but also many new faces who have never been here before. So we've been in business since 1999 as the first institute that was only focused on the biology of aging. And today we are, I guess, growing and prospering. As you know, we have about 300 employees here, 24 faculty members, all working on the biology of aging. And one of the things that really excites me about hosting the first Biomarkers of Aging Symposium here is the idea that this is part of a transformation that the Bach and the field in some way is undergoing, which is the idea that we're taking all of this biology and trying to bring it to humans. So welcome. I could not be more excited to have you here. As you know, this symposium is part of a whole week of meetings here at the Bach and at Stanford, and that I hope will become a tradition that we call Longevity Week. So we have this meeting today. Tuesday and Wednesday, we have a Longevity Summit. Thursday, we have BAM, the Bay Area Aging Meeting at Stanford. And Friday, we have the first symposium focused on all of the clinics that are emerging in the space of aging research. So without further ado, I have to make one brief comment about the fire exit, which are at the end of the auditorium. So in case we have a fire, which we've never had, so I hope it's not going to happen today. You know where they are. Welcome again. Vadim, you are the first.
Good morning, everybody. I'm just so happy to see so many familiar faces, and thank you very much for coming. This conference was conceived by junior people. You can see some senior people here, but actually these are the people who have done all the organization and arrangement and actually even conceiving the consortium. So first, I'd like to thank all of these people, and you will see them throughout the day because they're also helping here. The interest from the community was just amazing. I just couldn't believe, because we thought it would be like maybe 50 people, but in the end we had to accept 100 and then 200 and then 250, and it exceeded capacity, and then it's like 50 people on the waiting list. It's just completely crazy. So basically all of the leaders are here in the field. So it's good that we all meet and we discuss and address this really, really important problem in the field. I mean, both opportunity and problem. Yeah. So let's do it. So first, just a small announcement that at 12:30 we will have a photo. Yeah, just let's do it very quickly. Just at 12:30, just go take photo and then go for lunch, because there will be poster sessions. And that's it. So we don't have much time. There's a large agenda to discuss, and I'd like to introduce the first speaker. And yeah, he doesn't need, of course, introduction. So Steve.
Only for 12:30. That's the photo.
Steve here. Steve Horvath. Yeah.
Good.
Thank you, Vadim. Mahdi Moqri. Thank you, Chiara Herzog. Thank you all for organizing this event. I will talk about my favorite topics, epigenetics. I want to go back in time, 1988. There was a paper by Baker and Sprott on biomarkers of aging. And some of you weren't born then. I hate to admit, I can come clean to you. I was alive in 1988. So yesterday, I spent $51 for a PDF file to download it. So if you don't mind, let's actually read it, you know. So go to bullet point 3, the biomarker. So it's about criteria on biomarkers of aging. And what is bullet point 3? The biomarkers should have high reproducibility in cross-species comparisons of functional and physiologic age versus chronologic age, particularly within the same classes and certainly within the same families of species. What does that mean? To me, it means, and I really simplified these kind of criteria. Look at axiom 4. A biomarker of aging ideally should apply to all mammalian species, Well, if it doesn't apply to Drosophila, fine, we can live with that. But it would be nice if a biomarker applies to all mammalian species. And coming back to the $51 PDF, in that paper, they enumerate all these species and they point out differences in maximum lifespan. And the good thing is, fast forward to 2023, we now know that these very ambitious criteria can be satisfied. So, and they can be satisfied by biomarkers that look at cytosine methylation. And in a minute, I will talk about so-called pan-mammalian epigenetic clocks. So why did it take so long from 1988 till now to develop these biomarkers? Well, let's just think about it. Back in 1988, I would say all the conceptual issues surrounding biomarkers of aging had been fully worked out. It's a strong statement. We should ask Luigi Ferrucci about it, or others. But I would say there were phenomenal papers on biomarkers of aging back in the 80s. And more than that, in the 80s, actually in the 1930s, we knew the benefits of caloric restriction and even parabiosis. By now, these are gold standard interventions, but it was all known. And then, as I showed you, of course, we knew maximum lifespans of most species.
Hey, good morning.
But what was missing in 1988? First of all, we didn't have the human genome sequence. We didn't have array technology or sequencing technology. For me personally, relevant is we didn't have methylation arrays. We also didn't understand chromatin states, histone modifications, functional genomics. And then also what we didn't have was the right culture. So to me, the greatest change in my personal career was this open access movement. I remember I was a doctoral student, and there was one data set, and the person who created that data set ended up on 1,000 citations because everyone used that one data set, But we are also fortunate that our community, the biomedical research community, really embraced open access. I mentioned Gene Expression Omnibus, the Cancer Genome Atlas, GTEx, Gene Networks from Rob, and many sources where you get data without any strings attached. You just download them. And this brings me to my request for this year. And tear down the walls surrounding data from human epidemiologic cohort studies. It would do a world of good. And there's an inside joke here because this all goes back to 87, 88, Ronald Reagan in front of the Berlin Wall, tear it down. It can happen. And we even know how to tear down these walls. It's really generative AI. You take a data set from a human study, and there are severe restrictions about data sharing. But guess what? You can create synthetic replicas from these data, and they contain most of the signal. And you can share them, So I feel the whole field of biomarkers of aging needs that open access revolution. I want to briefly mention that I've always been very grateful about open access, and we just returned the favor by releasing data from our mammalian methylation consortium, 15,000 tissues, software code, you right click it and download it, no legal form whatsoever. Armin Hagani, who is around, can tell you all about how to get software and so on. So I want to now talk about these multi-species clocks that do satisfy the criteria by Baker and Sprott from 1988. And let me start out with a little bit of math. You want to define the concept of relative age. You take the age of the animal and divide it by the maximum lifespan of the species. For example, the rat, a 2-year-old rat, has a relative age of 0.5. Similarly, a 61-year-old human also has a relative age of 0.5. And Akelu will present a poster where she describes this universal pan-mammalian clock that applies to really all mammalian species. And on the right panel, you see how methylation age predicts this relative age, So it's really applicable to all these species. Here I show you how a pan-mammalian clock applies to humans, to mice, dolphins, and so on. These pan-mammalian clocks are equally accurate in long-lived species like a whale or an elephant or a human as in short-lived species, shrew, mouse, rat, and that is shown here. But very importantly, these pan-mammalian clocks also predict mortality risk weekly. They're not great mortality risk predictor, but they do predict it. And we know that from the Framingham Heart Study, the Women's Health Initiative, and so on. And also, many of you are working on OSKM, Yamanaka, interrupted reprogramming. These pan-mammalian clocks also respond to that. So there's another major advantage of multi-species clocks. Well, you can actually evaluate them in several species, especially mice. And therefore, you can do what I call test the test. You have a biomarker, and it better respond to high-fat diet in a mouse liver, or it better respond to caloric restriction, or in various dwarf mice, or parabiosis, do you see? So a multi-species clock allows you to leverage all these mouse studies. And this brings me now to a challenge for the entire field of molecular biomarkers of aging. We really need to move beyond cytosine methylation. It would be so nice to have pan-mammalian clocks based on other omics types. Histone modifications is the obvious one, proteomics, lipidomics, glycomics, RNA, whatever, or particular hallmarks of aging. And in this context, I was so pleased to see these exciting posters that are outside here that describe clocks based on histones, clocks based on proteomics, clocks based on lipidomics. Very exciting time, but it will be worthwhile to generalize those to also work in two species, human and mouse. Now I come to the topic of methylation clocks. What are open challenges? First of all, we want even higher predictive accuracy for morbidity and mortality. So yes, these clocks work beautifully when you look at several hundred people, but wouldn't it be nice to have clocks that are so accurate that they would inform us in an annual physical, We need clocks that are more responsive to rejuvenating interventions. We need methylation clocks that apply to blood, but they reflect organ function. And some of you are working on that. It's the idea of, is blood a good surrogate for liver or other organs? Then we want more interpretable clocks. The drawback of machine learning-based clocks is often interpretation. You don't know why do they work so well. And we want, of course, improved single cell clocks. We already have some, but yeah, let me just mention a few things. So I think everyone is fully aware of the power of artificial intelligence, of deep learning. And there's even a poster here on other machine learning approaches, ensemble predictors. So there are these wonderful approaches to increase the accuracy through deep learning. But again, deep learning requires very large data sets, very large human cohort studies. Think 100,000 samples, 500,000 samples. So maybe we can train these tools on synthetic data. Now I want to highlight another challenge, platform differences. Many of us use the so-called Illumina Infinium array. And there's, for the experts, it's called Epic Array. But now recently, Illumina came up with version 2 of the array. And we've been struggling adapting our biomarkers to version 2, you know. So this is kind of, to be honest, it's kind of frustrating, you know, that that could happen. But more broadly, forget methylation arrays, new approaches for lowering costs targeted by sulfide sequencing, or once the costs have been really reduced following Moore's law, how do we leverage whole genome bisulfite sequencing data or nanopore sequencing? So many technologies. We need ways to translate biomarkers that were trained in one platform to those on another one. And I briefly mentioned, again, our mammalian methylation array data. They were generated on a special array. But how do we impute these particular cytosine locations using human arrays? It's challenging. So the best thing would be to measure the mammalian array probes in a human cohort studies. It would be invaluable because we could import 15,000 tissues from mammals and make them directly relevant for human health. I'm not aware of a single epidemiologic cohort that has measured the mammalian probes. Then there's other challenges for methylation technologies. Something as basic as pre-processing, so-called normalization methods for array data. And Albert Higgins Chen, Morgan Levine developed a particular approach based on PC, principal components. And but Alexandra Binder, who is in the audience, will present another model as part of her poster on improving the reliability of epigenetic clocks. We call it the tuning method. So I invite you to talk to her. I think it does a world of good in reducing technical noise. It's just bioinformatics. Another challenge is what one could call causal clocks. Everyone says we need causal clocks. Well, how do you get causal clocks? One approach is longitudinal studies. And Dan Belsky will talk about his PACE biomarker. But fortunately, there's already quite some literature here of using longitudinal data one way or another, you know. And so that's so valuable. Another challenge, as I mentioned, is this question of understanding the biologic mechanisms underlying epigenetic aging effects, or conversely, what are the functional consequences? And you see a couple of posters here that will be presented later today. Somatic mutation could be an explanation of epigenetic aging, or various structures of DNA and RNA. And then we should also highlight the clock from Mahdi Moqri, Vittorio Sebastiano, and others, which they call polycomb repressive complex 2 clock. It's a clock that only looks at a particular chromatin state, bivalent regulatory regions based on PRC2 binding. I think these, what's good about it, well, directly interpretable, you know, Yes, but similarly, you could build methylation clocks based on enhancer regions or methylation clocks based on repeat elements. Or, you can carefully think about what regions you want to query. So this long-standing criticism that we don't understand the mechanism of epigenetic clocks is a little bit outdated. Many people have really spent the last 10 years trying to understand mechanisms. So I mentioned here a paper from Ken Raj, who has done a lot of in vitro studies, and Martin Picard's team. But many others have really used fibroblasts, UVECs, many keratinocytes, really to study hallmarks of aging, anything from mitochondrial function, DNA repair, and so on. I would argue we know a lot about the mechanisms of epigenetic clocks. And coming back to this very difficult topic of how do we understand causality? One approach is to use genetics, Mendelian randomization, but more generally. There are lots of, a lot of effort has been done. In the bottom, I show you an article in Genome Biology by Riccardo Marioni, where we looked at really, I want to say 30,000 people to build polygenic risk scores of epigenetic clocks. But here you see also at this conference, there are wonderful posters where people use Mendelian randomization one way or another to dissect causality. Yeah. And the other part of genetics is mouse genetics. There's so much we can learn from these dwarf mice, for example. So this is work from Richard Miller, and Vadim Gladyshev was the first to show that really most of these dwarf mouse models show very reduced epigenetic age. And more generally, the literature is full on also human genetic conditions, Down syndrome, and the so-called overgrowth and undergrowth disorders, Soto syndrome, Tetoon-Brown-Raman syndrome. So we have a pretty good understanding which mutations affect epigenetic age. And now I want to come to a conceptual challenge, and it's a psychoanalysis of the whole field of biomarkers of aging. Half the room is what one would call lumpers, and I'm Mr. Lumper. So I get excited about a pan-mammalian clock that applies to all tissues, applies to the entire life course, from prenatal age to centenarians. I lump. But then there are the splitters among you, and I still talk to you. But these are the people who will focus on one cell type, stem cells, one particular species, humans, one race, Hispanic, one sex, females, one age group between 40 and 60, and you will build a beautiful clock in that very limited domain. And just a brief shout out to Nia Einon, who traveled here all the way from Australia to visit us, but his team developed an epigenetic clock for human skeletal muscle and then showed that exercise rejuvenates it, So I do think there's tremendous value in really these very targeted biomarkers, So coming back to epigenetic clocks, they must be judged by the criteria of aging. And above all, they need to predict future onset of age-related conditions. You've heard me talk about Grim age. It's the most predictive biomarker that came out of our lab. Arkelu developed that. But there are by now several other mortality risk predictors. I invite you to look at the literature. And many people have validated Grim age. And I'm very excited that there's a poster that looks at lifestyle factors on how does it affect Grim age. So validation. And here I show you how grim age allows you to stratify mortality risk. These are now different survival curves, so-called Kaplan-Meier plots, in different cohorts stratified by race and sex, and you see pretty good divergence. Okay, I stop with epigenetic clock theory of aging. Maybe aging is a software design flaw. Something goes wrong in development. And there's a wonderful poster on prenatal influences on epigenetic clocks. I think it's exciting, you know. So aging may start in the womb. I started a non-profit, if you want to, but there are many other vendors of clocks. And I'll stop with that. Thank you. Mm-hmm.
Thank you very much for your visionary talk.
Thanks. Mm-hmm.
First, there's a lot of people on the back. There are seats here front. Just move here instead of just standing. And second, somebody parked an Airstream van number 62675L3. Could you please move it from another parking lot? Thank you. So now we move to the first panel. So as you know, this is a conference of the consortium, Aging Biomarker Consortium, and we have a panel with scientific chairs of this group. So Andrea Meyer, Vittorio Sebastiana, Mike Snyder, and Eric Verdin. Could you please go on stage? Well, I think we will have a nice discussion. So I just want to introduce just very briefly, these are people who chaired the consortium in 2023, and they could provide insights into how the group has been organized and what is it about. But I first wanted to ask you to introduce yourself and maybe state a concept very briefly, just what you think is most important in the field in terms of biomarkers of aging. Eric?
Good morning. I've already said good morning to many of you. I'm Eric Verdin. I'm the CEO and the president here at the Buck Institute. I also run a lab that's been working on epigenetics and immunology and the biology of aging. And I enjoyed Steve's presentation because I would consider myself one of the splitters. And so one argument that I would make is that I think the understanding of what these clocks are really doing biologically for me, at least from what I've seen and read, is really fragmentary. Yeah, there are some hints, but for many of the sites, we don't really have a full understanding whether they're truly causal or they are sort of carriers type of sites. And so some of our work actually has been really focused on the blood, which is the compartment that many in the fields will access as biomarkers of aging, and trying to understand what are we measuring when we're doing the epigenetic clocks on whole blood. And I think one of the findings that we've made is, even though there have been effort in the field to deconvolute variations in the relative abundance of different cell populations, there are more than 50 different cell populations in the blood. And we know that they change during aging. And we've been able to show simply, for example, if you compare naive T cells versus terminally differentiated CD8 T cells, you have a 20-year difference in age. And so this is likely to really have an effect on the numbers that we generate. Independently of this, when we look at naive T cells of people of different ages, we still see a trace that's linked to aging. And I think what this tells me is that the clocks, when they are integrated into a whole organism, or whole organ, as the blood can be considered, are actually a mixture of multiple biological events. And I think our effort is really trying to understand what are these events. And we've generated a clock that's called Intrin clock, which clearly measures something different from the standard clocks, which I would argue actually are highly biased towards inflammation and chronic immune activation. I think that's pretty much
okay. Thank you.
Where,
thank you, Eric. Andrea? Yeah.
Hello, good morning, everybody. I think if we see who is in which tribe, I think I'm in Steve's tribe. Sorry, I bring the splitters together. I am an internal medicine specialist and geriatrician by training. I'm directing the Center for Health and Longevity at the National University of Singapore. We do lots of randomized controlled trials to see how we can translate geroscience hypothesis into humans into interventions. So we are focusing on lifestyle interventions, lots of supplement trials, but also repurposed trials with an age group of 40 to 60 years. Sorry, there I'm a splitter at the moment. So we are really focusing on healthy individual and really seeing how we can optimize the health of individuals. In the trials, we are even now stratifying our individuals based on biomarkers of aging. Think about epigenetic clocks. We have all 11, which I think are quite valuable at this moment in time, integrated into our trials to really see what kind of changes we see. What we really would like to achieve is linking the biomarkers of aging together with appropriate interventions in humans, that we are going into a new arena of healthcare, and that's precision healthy longevity medicine in the end. We also, and then I'm stopping, we also now bring what we know in geroscience and health longevity medicine into clinical practice. We opened in Singapore the first publicly funded longevity clinic at the 31st of August, which is now up and running. And we also have private clinics there where we gather all the data. I think data are very important to validate all the biomarkers of aging. And I think the last point I would like to make is we shouldn't restrict us to the biomarkers of aging, which are biological, but also integrate them to the clinical biomarkers, and especially the digital biomarkers of aging. Thanks.
Okay, thank you, Andrea. Mike?
Okay, I'm Mike Snyder. I chair the genetics department at Stanford, right down the road. I also direct the Center for Genomics and Personalized Medicine. I'm mostly a splitter, occasionally a lumper, but for the most part, our lab studies uses big data to track health and wellness, and we've been doing this for quite some time. I'm a believer that you should try to hit markers that are actionable, so I like that concept, although understanding ultimately leads to action as well. So I guess in that sense, I'm a little broader than that, perhaps. Anyway, I'm a believer that we should be able to reverse our age at some point, and I hope we can do it soon before I expire.
Good morning, Vittorio Sebastiano. I'm a faculty at Stanford in the OBGYN department, and I'm also a co-founder of a company that is trying to implement epigenetic reprogramming for clinical interventions, term bio. Well, if I were to define myself, probably I'm a splitting lumper, meaning that I obviously overall understand and value the fact that there must be some commonalities, when it comes to aging, but I think that at the same time, there are some human-specific traits and human-specific features that I think need to be kind of dissected, and obviously being specific, they require specific types of tools. So that's my opinion. I am personally excited about the fact that, I'm excited about the development of causal clocks for a very simple reason. First of all, because we might really be able to understand what is at the epigenetic level causing aging, and I think that really gives us an opportunity to develop drugs, biologics that could reverse those traits and really bring back the biological clock from a functional standpoint in different cell types and tissues. I think we will elaborate more on
We will
during the discussion.
So, and then maybe I will introduce myself as well as an additional chair. My name is Vadim Gladyshev. I'm a professor of medicine at Brigham and Women's Hospital and Harvard Medical School, and we work on biomarkers of aging. I think that the ability to quantify biological age has been a major advance in the field. Thank you, Steve, and everybody else here, so for contributing to this collective effort. And we've been developing various biomarkers in model organisms in humans. We are very interested in causal, as many other people here in the audience. And I particularly like the aspect of using biomarkers to understand the nature of aging, because ultimately I think we really need to understand what is our enemy so that we target it most efficiently, and biomarkers kind of help in that direction. So this brings me to the next question to the group. So what do you think is the best biomarker, or maybe it was a good biomarker, or how do you feel, what should we use? Anybody?
Well, I'm a believer you want common biomarkers like Steve was espousing, because that way you can compare your results across laboratories, across experiments, across systems, and that is valuable to know how things are going systematically. And I like to use a car as an example. It's nice to know how much rust is going on at all parts of the car, what's going on there. But at the same time, here's where the splitting kicks in. I'm a believer that having biomarkers for specific areas makes it more actionable, makes it more valuable. And there, I like to use a hearing aid analogy. When my hearing goes off, the rest of me might be going pretty well. I do have a hearing aid, by the way. It's a sensor, of course. But it basically lets me fix that particular part while I still try and understand everything else that's going off with me as well. So I like having specific biomarkers. So it really depends on the application. So when you're trying to fix something or study some particular area, like the muscle example that Steve used, that's very, very valuable. But at the same time, if I want to compare what's going on with muscle with all the other things going on or other laboratories, you do need common. So I'm a believer in both.
Maybe I want to sort of emphasize the point that Steve made on the need to move beyond simple epigenetic clocks, because there is evidence for clocks at the level of the proteome, lipidome, metabolome, and so on. And I think just want to highlight the fact also that we've joined force with Lee Hood, who's here, who's going to be speaking in a few minutes, with this Phenome Health organization, which is actually looking at all of the omics and building a platform that is going to be helpful in actually integrating all of these omics. And I think we're quite excited to have Lee with us, helping us to build maybe some new multifunctional clocks.
Yeah, thank you. Jay?
I think we have to consider two things. The pace of aging is quite different organ systems within individuals, not only between groups of individuals. And that's what we normally try to achieve to compare groups of individuals. But if you're looking and you have rich databases within an individual, the pace of aging is quite different. And the mechanisms of aging might also be very, very different between the organ systems within one individual. You have the pathophysiology and you have the pace. So there was, at least in clinical practice, now we apply all the biomarkers of aging or what we can do in different systems with different body fluids to get an understanding about the dashboard, I would say, of our car, and actually showcasing us where the weakest link might be or where mechanisms are to interfere with. So I think pace and origin, that's what we have to disentangle in the next decades.
Okay, thank you.
On years.
Vittorio would like
to
add?
Also because I think at some point, I mean, relatively shortly in the future, in my opinion, so I'm the half full cup guy always. I think we are really going to get to the point where we will be able to rejuvenate, let's say, the liver or the heart or the brain or whatever that is. So obviously there's going to be an intrinsic rejuvenation factor, right? But then what does that do systemically to the body? So does that mean that rejuvenate? And once we will understand, then maybe there are 1 or 2 or 3 key tissues that age. And because of that, for example, the hypothalamus, because of that, the whole endocrine system is aging. Now, if I can rejuvenate the hypothalamus, of course, I have a juvenile hypothalamus, but that actually potentially could have systemic implications. So does that mean that the biomarker now that I have in hand can capture that systemic, or it's not? And so, I think it's very important. So I don't have an answer to your question. I don't know what, a good biomarker should be able to capture all of these. Does it have to be a lumper or a splitter biomarker? I don't know, but yeah.
I think this highlights some of the open questions, right, in the field, that we don't really understand exactly how aging of one organ influences aging of other organs, what's systemic biological age, and these are the open questions, I think, for the community to solve in the future. So I wanted to ask you a little bit to elaborate a little bit more on how you use biomarkers in your lab, labs. What exactly questions you address, and what's your current approach to biomarkers? Anybody? Eric, would you like to start?
Yeah, maybe I already started actually on my first intervention. I think we study aging in the immune system, and mostly in the human immune system, which we think represents a really readily available organ that you can sample, and you can intervene, you can modify, and so on. So I think that's through that work that we ended up finding out that as an organ, if you think about the blood as an organ, it is highly heterogeneous, and each different cells show very different age. And the question is, what does that mean biologically? And what are the implications of this in terms of in terms of immune aging, basically? That's where we are.
Okay, thank you. Andrea?
Yes, so we have different approaches in my lab at the moment. So we are using big data sets like the UK Biobank to really showcasing how we can make use of all the data to establish new clocks, but also validate others. So we have shown that the organ age is quite different in individuals, and that we can actually predict possibly what the weakest link is in the organ systems within individuals, and what kind of sequence we see in the aging process. But we also are looking at very specific groups of individuals, for example, patients with delirium or undergoing surgery, to really see what kind of methylation changes we are seeing in blood samples. Most of the times we have in these individuals to see if we can improve clocks we have already. I think very excitingly in the intervention space we are doing in our lab, or the RCTs we are doing, that we are now risk stratifying individuals based on biomarkers of aging. So normally we would say, okay, we want a female 40 to 60 undergoing that and that intervention. Now we are using epigenetic clocks to include only biologically older individuals in our trials to hopefully rejuvenate their biology. So it's lots of applications at the moment and validation to see what we can introduce into clinical practice.
Thank you. Mm-hmm.
Well, many of you may know we do multi-omics profiling, deep data as well, do a lot with digital markers these days, hence all the wearables. So we try and use all sorts of data to try and track people's, if you will, aging patterns. And I do think we need all those things. And if you look at how people go off, I think we are, as humans, we're complex homeostatic systems where there's an interplay between our metabolic and our immune and all these. I mean, I think we all know that. And you have to keep that in balance. But what can easily throw you off is these stochastic events. We all have something like, if you start looking at data now, 30 million pre-cancer mutations, all of us. So at some point they can go off, and let's face it, we'll all get cancer. For anyone living for 100, it's going to get cancer at some point in their life. And so the question is, how does that get cleared? How does that, get handled? And so I think we need markers that detect these sorts of stochastic events, as well as the general sort of rust, if you will, or have just going on. So I do think we need specific systems. I think we need to keep our homeostatic systems in check as best we can so that we can avoid these stochastic events. Again, the other area where most people die right now is cardiovascular disease. So that's can be a pretty stochastic event, severe one as well. So I think we have to somehow be on the alert for those as well.
And in my lab at the moment, we're looking at the whole genome methylation data sets. And the reason is very simple, because since we're dealing with epigenetic reprogramming, we do see that epigenetic reprogramming transiently applied impacts pretty much every hallmark of aging. So from transcriptomic to senescence, mitochondrial function, and so on. So that, to me, speaks for a massive epigenetic resetting of the cells. And so the epigenetic clocks that are currently available, and we collaborated with Steve, and that was a fantastic, because the clock clearly showed rejuvenation, when we used Steve's clock. At the same time, though, only one cytosine was showing because it had the biggest weight in the analysis. And to me, I mean, when I started looking at this problem, I said, it cannot be that just one cytosine out of 7 billion can explain this massive rejuvenation that we're seeing. And that's why I was kind of driven, together with Mahdi, driven by this kind of urgency of understanding the biology behind that. And we developed the index and we see that this is phenomenal because, I mean, I don't want to publicize, I mean, so what we saw was that there are specific regions of the genome that are gaining methylation with time. And what's really interesting about that is that if you look at different rejuvenative intervention, rapamycin, caloric restriction, epigenetic reprogramming, they do different things in the genome, which is fascinating because, and that means that they all work to some extent, but we need to understand how they work and what they do, because then we can really develop then the appropriate biomarkers to define efficacy.
Yeah, thank you. So one reason, like we have so many people here in the audience and there's much interest in the community is just because this development of biomarkers provides opportunities to quantify aging, but also we have many challenges. So I wanted to ask you what you think are the main challenges in the field. What are the open questions that we face and what could be potential solutions to these questions? Anybody? Just what is the key kind of in the field? What should we do?
In my mind, I think everyone recognizes that as we move to a translational aspect of aging, we're going to be looking at interventions. And everyone recognizes that we're not going to be doing 30-year clinical trials. So the excitement about the biomarkers is the idea that they're going to be proxies for determining or predicting the efficacy of an intervention in terms of longer lifespan or longer health span. And I think, you know, there are markers, some of the markers that we have do this, but we've heard, you know, very imperfectly. You know, for example, the fact that the clocks are behaving in different ways or responding in different ways to interventions that all you know, prolong health span and lifespan, to me speaks to the fact that there are multiple mechanisms and we really don't have an understanding of this. So I think for me, the focus of this field should really be on understanding and developing the best predictive biomarkers for longevity that responds to interventions.
Okay. Yeah. Thank you. Andrea, do you agree?
I agree. Yes, we often agree. I think the surrogate endpoints are very important because in the end, we also have to really not convince, I would say, the regulators, but at least be in speaking terms with them to have FDA-approved outcomes in the end. And this should not just be a 6-minute walking test or hand grip strengths, etc., but really showcasing that we can influence the biology of aging. And I had another point, yes, and that's, I think, the combination and the composite biomarkers of aging. So how do we really understand the individual biology of one person to then target that biology in a much more in a precision medicine way? And of course, I know precision medicine is treating groups, but in the end, hopefully it's as one group. So I think that's very important. And there are lots of companies doing that. It's machine learning, building digital twins. We also do that at NU. At the company we have, I think it's very important to get the understanding. And I just wanted to highlight Steve's comment, how important it is to bring all the data together and makes them accessible and easy accessible. I think if we can achieve that, I also struggled in the past to get even access to data I established myself. Let's get together and bring this huge number of data together to learn about what the beautiful biology really is. And we have so many freezers which are untapped. So let's do that.
Okay. Mike, would you like to continue? What are
Sure.
the most important? The most important
actionability. And short-term actionability, I think, is solvable. Again, fixing very specific, say, muscle or what have you, that should be less of a problem than systemic things where you're trying, like you said, rapamycin to show has effect on multiple systems and long-term consequences. So I think that is a challenge. It also doesn't fit well in the US with our healthcare system. That is, it is sick care, not healthcare. And so nobody really pays you to run trials that will keep people healthy for long times. The financial incentives aren't properly aligned in the US for this health system and running trials that will keep people healthy. So I think we should figure out ways to better implement that so it winds up getting done. Right now, it's done in a very grassroots fashion. Maybe that's how it has to be done, but I think we could do better.
And to me, two things very quickly. The first one is the one that we mentioned together, a couple of days ago at the Foresight. I think that's the academic research needs way more funding actually to develop this type of knowledge. Unfortunately, this is the classical fishing expedition type of grants, very boring, very descriptive, the one that NIH usually doesn't fund. But I think we need that type of research and the resource, because there's still a lot of knowledge that needs to be developed. And I really strongly advocate actually for making sure that this happens. The second piece is this. I think that kind of piggybacks on what was just said by Andrea. It's like sharing information, putting together the tools, kind of building a consensus, and really speaking together as a group of people that are about the challenges. Also, because at some point this whole effort needs to go to the FDA, because the FDA will have to implement some of these biomarkers. So how soon do you reach out? What are the biomarkers that you're gonna propose? And so kudos to Mahdi, Jesse, and all the organizers of this conference, because I think this is exactly what's needed. And I hope actually that more events like this are going to come to really bring together the opinion leaders and actually new people to really bring new ideas.
Yeah, go ahead.
Yeah, it's great all points from everyone. I just wanted maybe to add one more, because it's one that is, I guess, close to my heart as a physician as well. There's a whole part of this field which is occupying itself by trying to make aging a disease, and they're fighting a battle which I think is fruitless in a way, because it just creates a backlash of reaction. If aging is a disease, that means everyone who's above 30 in this room is actually sick, which does not work with physicians, does not work with regulatory agencies, and it's not necessary. And so in some way, what I think the field of biomarkers can provide is a measurement that actually determines where you are in this aging process. And that measurement itself will be the indication. And there are many precedents for this in medicine. If you have high blood pressure, you're not sick, but you have a marker which is indicative of potential problems in the future. And I think, this is where our field actually could really be guiding the whole field of interventions, is by providing something like a high cholesterol level. Again, that's not a disease, but it's considered something that's necessary to intervene with. And I envision a day where we will have a marker that your doctor will measure and will determine that this is an indication itself for clinical trials for rapamycin or metformin or any other intervention. So I think, to me, this is where we can sort of get out of this debate: is aging a disease or not? It doesn't help us, at least that's my perception in the general audience.
Thank you. So let's now move to the question of the consortium. So this group was formed to bring together a community to provide a platform for the discussion, on open discussion of major challenges in the field. So how do you see the future of this group, the future of the consortium? What should you do? What kind of activities we should pursue?
A lot more of these meetings. And I think, I like the point that Vittorio made about the fact of advocacy. I mean, organizations like this, once you get to a critical mass, and I mean, the fact that we have a full room. I remember when we talked about organizing this meeting, I said, how many people are going to come? And we thought maybe 100. And, we had actually to close registration, which is really amazing. So that really speaks of the fact that most of the people in the field actually are getting it, how important it is. It's going to be the rate limiting point in terms of advancing this field. So I think for me, more meetings, more coordination, more advocacy for funding, that would be.
Yeah. Okay, Andrea?
I think the first very important step was what is the definition of the all the words we are using. So what is a biomarker of aging? So that paper has been published. Kudos. So I've huge applause, I would say, to everybody sitting there doing the really great work. I think that's very important. And now using this terminology would be great. So if everybody can use the same keywords and putting them into the papers, I think it's very important. Yes, let's give an applause for everyone. So, and there are other papers now in the making. I think that's very important. And I think what we also should do, being very inclusive, to invite everybody that we have tough discussions. I missed that a little bit in the first paper, having these rough and tough discussions, but that was in the backyard, I think, in the engine room. So I think that's most important, and then sharing data and talk to each other to see if we can work together. I think that's most important. Let's see where it ends. And of course, I would love to see the validated markers being implemented into clinical practice and having these big data sets to make them even better.
Well, I'm a big fan of common markers and specific markers, coming back to the earlier discussion. So I like the idea of getting people to use a reference that everybody uses. If you're doing a sort of standard set of experiments, say, go ahead and use the methylation clock, or we set on a few clocks. It doesn't stop you from doing your own specific assay, which I think you do need to solve the problem you're doing. But I think if we can get references done, especially in large studies, so that we can share data and compare data better, would be very useful. I'm a big fan of the data sharing too. I think maybe there's more for the field itself than necessarily the biomarkers of aging, but there's 8 billion people on the planet. And even if you shared 1% of that, that's an incredible amount of data. Easily 1% of people would make their data completely open. Right now that is hard. So picking up on some themes that were mentioned earlier. And it's not really the people who don't want to share the data, it's the scientists, let's face it. They really hoard their data. Try and get wearable data or CGM data these days. It's just impossible. It always says in the paper, data is available upon request. We request it every time, and we almost, in fact, for the wearable data, I don't think we've ever gotten it from another group. And that's just crazy. So I think this group, whether it's a biomarker group or the larger group, could easily figure out how to organize data. The citizen science movement is pretty strong. One could easily figure out ways to try and do this in a more organized fashion, more systematic fashion. So you can pull out data across literally 1 billion people. I think it's very, very doable. That we could follow what's going on. Then you can look at environmental impacts, all sorts of problems that we're just not touching right now. So I think we could take this to a whole new level.
Well, they covered pretty much everything. The only thing I would like to say is that let's also make an effort to keep an open mind, in the sense that I think over the past 50 or so years, one of the quote unquote issues about the aging research in general was that people were really taking a very narrow-minded approach. Everybody was a fan of a particular hallmark, and they were just looking at that in singularity. I think this is not a singularity problem. This is a broad problem. And we now historically have the opportunity, thanks to the technology, omics, AI, and so on, to really put together large data sets coming from this disparate kind of, domains. And so I think it's we need to keep an open mind in that sense, that we really need to also always keep an eye for new approaches, new ideas, new people, and not keep it as a kind of a club of people that think the same. Because if we keep thinking the same, we're not going to have any progress. We need to be open-minded and open to something new. So that would be my.
Yeah, excellent point. Thank you very much. So let me ask you now just a few questions, like a little bit on a more specific manner. So for example, like Mike, you pioneered this concept of agiotypes. How do you envision that concept could integrate with more kind of generalized approaches to aging? Because ultimately we would want to work at the population level.
Well, I think population level is nothing more than an aggregate of individuals. So I'm a believer, everything comes down, I hate to say it, to yourself. It's maybe a Donald Trump sort of view. But Yeah, I think we want to, it's nice to have cross-species things. I think that's nice from a testing perspective, but at the end of the day, you want actionable information, and actionable information comes down to the individual. So I believe that you want to track individuals, and we all age differently. That's something we and others have shown that, and you want to see what's going on. And sure, it's nice to come up with systematic solutions, but at the same time, you want to have very specific solutions for solving the individual problems that are arising. And also, I think if we understand how the individual problems are arising, we can understand the factors that are leading to those, and then try and mitigate those, if not in our generation, and future generations. So I think by tracking individuals and their age types, we can again get action at the individual, and then you just aggregate the information to get population level understanding.
Yeah, thank you. Vittorio, what is your opinion on the use of biomarkers for anti-aging interventions, longevity interventions? Are they usable? How should they be used?
Yeah, no, I mean, that's a critical point. As I mentioned before, I think that currently the biomarkers that we have do capture, they can measure rejuvenation, but again, they do not really capture the biology behind what is happening, which I think is critical. And I made that example before, that actually different types of interventions are actually acting on different parts of the genome. And we also, because I envision, personally I envision the future where there's not going to be just a one single rejuvenative intervention. There's probably going to be a combination. Senolytics combined with reprogramming, or calorie restriction combined with some dietary intervention. So again, each one of those is going to have an effect, but the combination of the two or the three or the four are going to have a different type of effect on the genome. And so we need to understand that. We need to absolutely understand it. And currently, again, the clocks that we have are great tools to measure chronological aging and how we are far from that, meaning biological aging. They can capture rejuvenation, but they cannot capture the biology behind it. And I think we really need to understand the biology behind all this process.
Yeah, thank you. Andrea, you use biomarkers in the clinical settings with various populations and biobanks. What is your kind of insights in this? What is the best practice to use them?
I don't think that we have a best practice yet. So, but this is what we try to achieve within the Health and Longevity Medicine Society. If you haven't signed up yet, hlms.co. So that's a healthcare professional society bringing really like-minded and also the non-like-minded individuals together with three aims to writing guidelines. So how do we implement, for example, biomarkers of aging into clinical trials, or especially into clinical practice? What is ready to use? And secondly, really education. So what is a biomarker of aging? Next meeting, Steve will educate the clinicians how to use, for example, epigenetic clocks. And thirdly, to really build the basis for a trial network. And I think that's very important. We are all splitters, I think, in lots of research, what we do, but in the end, we should learn from the endocrinology of the IC unit world, where lots of trials are being done, even worldwide, decentralized. And that's what we try to achieve, to really bring the clinician scientists together to do trials and to implement biomarkers of aging, and there was be able to test if they're working or not. I think another very important point, which I missed in your last question, what we have to do, what we should do as a consortium, I think a standardization that if we are measuring a biomarker of aging, that it's reproducible in another lab. I think that's very important, because whatever we do in longitudinal studies or in clinical practice, we want longitudinal data, because then we are learning from our biology. So, and having reproducible biomarkers is most important, which are showcasing what the biology is. I'm not sure if you can measure mTOR or NAD levels, etc., from human beings, but I can tell you it's not reproducible with any lab I'm working with at the moment. So I think finding there the protocols how to do it is of huge importance.
Yeah, thank you. And Eric, so you focus on the immune function and inflammation. So how would you advise people to use biomarkers in that system? Or how do you use it?
So I mean, we don't know yet, and that's why we've been spending some significant time, you know, really dissecting this. I think my vision of what the field could become is, you know, when you start from and the blood again offers a really unique setting in terms of this is the only organ that's available in human for analysis in a very detailed manner. And as I mentioned, you know, there's close to 50 different cell types, which we know change during aging. I think as a working tool for biology, I think it's really remarkable. There's a lot of technology that's being developed that allows you now to subtype, you know, to purify these different cell populations. I could see maybe in the future, analysis could be done on different subtypes as a reflection of different, you know, different processes, basically. So I think that's, it's clearly, in my opinion, it's not ready for prime time. And, you know, one of the arguments that I've made why it's not ready for prime time is that if you, there have been a number of papers claiming age acceleration during COVID infection. I mean, I still remember one paper that claimed 15-year acceleration of aging by a COVID infection that takes 3 weeks. I mean, from a biological standpoint, it just doesn't make any sense to me that you would age by 30 years, by 15 years over a 3-week period. Now, when you start looking at what this data meant, it's just the clock was measuring a shift in populations, massive immune activation, which means much less differentiated T cells and much more differentiated T cells, much less naive T cells. And if you know that the naive T cells are 20 years younger than the other ones, but there you have your phenotype. But the same thing, you know, can also be seen. The same point can be made in terms of a paper claimed the first rejuvenation of humans was the TRIM trial, in which humans were given human growth hormone and metformin with the hope of inducing thymus regeneration. And I think they were able to induce some degree of thymus regeneration, but what we know the major effect of this is to increase your pool of naive T cells. And of course, if you increase your pool of naive T cells, you're going to, and you look at the whole blood as an organ, you're going to have this impression of rejuvenation. It's not, at least it's rejuvenation of the thymus, but this is not sort of backtracking your aging process. So I think these data for me have implications not only from understanding what we're doing, but also in terms of the message that we're communicating. And I think a lot more work needs to be done. I still believe in the long run, the blood, its cells, plasma, serum, and so on will be the preferred area. This is what pretty much all of medicine is based on, at least most of medicine is based on plasma, and there's a lot of information there. So our effort is really to try to understand this better.
Yeah, thank you. So now we have a couple of minutes for questions from the audience, because this, again, so this is a group, I mean, there is no, it's an opinion, right? So I already see one hand, and so maybe you just introduce yourself.
Anyhow, I don't need much of a microphone. I'm Kevin Parrott. I've had the benefit of being associated with the Buck in the past, and still associated with the Buck. But my whole purpose is to minimize time to the development of interventions in aging, because I was a cancer survivor wondering why things were so slow. So the actual development of health technologies, I want to speed up. And in order to do that, you need clocks, because that's time, right? So Craig Venter in 1999, I think, or 98, threatened to patent the human genome, which caused the whole scientific community to gather together to create the Human Genome Project. Is that time now for scientists to come together to create a human multiome? Also, there's a collaboration aspect. We need a W3C standard, the Tower of Babel facing all of the scientists just trying to share information. Even if you want to, you can't. The NIH has made some aborted attempts to try to do this. but maybe we have to do it ourselves. Anyway, the human multiome, I'd love to see this group. Oh my God, the center of gravity. I can't believe it. 12 years ago, we had a similar meeting. We had 30 people. You know, it was amazing. Thank you, everybody, for all your work.
Yeah, thank you, Kevin. Yeah. Maybe another quick question or comment. Anybody?
Hi, my name is Mahmoud Panjwani. I'm from right here in Silicon Valley. So to my non-scientist, non-biological, non-medical, but startup mind, this is a systemic problem, obviously. We are the most complex system that has ever been created. And so to take from Steve's terminology, lumpers or splitters, and to Vittoria's point, I think we are either a splitting lumper or lumping splitter. It's a top-down, bottoms-up reconciliation problem, to me, it seems like. And since every human body is very unique, we are the most complex, unique systems that have been created, I think the approach where we are looking for one magic marker or one magic thing is probably not going to be easy. It needs to be looked at it from a systemic splitting lumper or lumping splitter, bring it together. So my point is that why not do a massive baseline for every human, right? Obviously that's costly. And then say, okay, if we have it, and if we have a bunch of thousands, tens of thousands, hundreds of thousands, millions of these data points, now we know what works for who uniquely, which is kind of the precision medicine that we are talking about. So just thought I'd lay that out as a non-scientist person. Like, I'm obviously not a trained scientist, so, but this is to the untrained mind here.
Right. Well, thank you for your insights. I'm sorry, Steve and everybody. So we need to move on. So thank you very much. Thank you very much. So it's just the program is very tight, so we just need to stay on time. So our next speaker is Luigi Ferrucci, and he will continue this theme of caveats in aging biomarkers. And Luigi is a scientific director of the National Institute on Aging.
Hello, thank you, Vadim, for the introduction. I'm really so pleased to be here. Having listened to the introduction, I want to completely change my slides, but of course it's too late. And you know, when I went to NIA for the first time, it was 1988. Many of you were not born. I visited, and the first person I talked to was Andrea Lacroix, which has been you know, really a titanic figure in the epidemiology of aging in the past and will be in the future, I think. And what she told me is that the problem of study aging is that we only have chronological age. We had something that will tell us about the rate of aging of an organism. We could do so much more. And I think that is unbelievable that we're almost there. I think that you know, I want to say the glass is full, and this initiative of the Biomarker Consortium goes exactly in the right direction. It's like the democratization of this discussion to those who really care, and clearly there are many, many people who care very much about it. So today I'm going to talk to you about some caveats, because I don't need to talk about the biomarker of aging. I think the introduction of Steve was absolutely perfect. So what are the things that I'm in mind that make me not sleep? I correct that. I sleep beautifully. I think that that has been, you know, always the characteristic my entire life. But let's say that worries me that fuels my thinking. One is, why do I need biomarkers? And we've discussed that, but I think that there is an aspect that we can concentrate on. And then the final problem is that I think we need to make sure that these biomarkers are sensitive to change. And I don't think that there is so strong evidence in the literature so far. And this is so important for what we want to do. We need to talk about underlying mechanisms. And I think that I agree with Steve that there is some emerging data that are important in this field. And then the relationship with other biomarkers and the hallmarks of aging. You know, what the other hallmarks have to do with it. And then I want to talk about, do we really need to work one biomarker across the lifespan? You know, our body is constantly changing, adapting to reach this incredible goal of longevity, and the mechanisms implicated are probably one after the other, subsequently, you know, it's not around, you know, there are testimonies that are given to different mechanisms across the lifespan. And I think that that's something we need to think about. So the way I think about aging, when I try to operationalize aging, is in terms of three basic domains that are strongly correlated. And the domain that I really like to focus on is always this one, that it's the functional aging. Because my goal is to say that people should be able to walk, think, and enjoy life as long as possible. That is our goal, but we know that in order to do that, we need to look at the phenotype that are underlying these functional changes. So the functional changing are here. I don't know how to use this. I hope I can show you something. Maybe I can use this. Yes. So, and for example, muscle strength or conduction velocity, and we have really championed this in the Baltimore longitudinal study of aging and in many longitudinal studies. However, we also know that in order to do that, we need to go deeper. We need to understand what are the biological mechanisms implicated in the decline of this phenotype. This distinction to me is incredibly important, is the baseline for any type of reasoning. And the reason is that life is characterized by compensation. So we do know that you may have really important biological changes that occur without any effect on the phenotype. You know, for example, you have millions or thousands of DNA damage that are constantly repaired. And until the compensatory strategies are effective, you don't see anything at the level of the phenotype. But at some point, the threshold of the balance between accumulation of damage and compensation is tilted. And so you start seeing phenotypic changes. And at the same time, phenotypic changing not immediately translate into functional changes. You need 30% of your muscle strength to have a normal walking speed. And only when you get below that threshold, you start seeing decline in walking capacity. You need 50% of your synapses in the brain to be able to have a normal memory, because our body has been created and built very redundantly. And because of that, we are lucky because biological aging provides you a window of opportunity of understanding who is declining rapidly at the time when you still don't have, you know, the phenotypic manifestation and the functional manifestation of aging. It can be detected at the time when probably the organ is more susceptible to intervention and more likely to recover, because the compensatory strategy, what I call the intrinsic prevention strategy that are already in our body, are still highly functional. Okay, second issue. Biomarker of aging, I worked with Steve for many, many years. And, you know, we created the Incanti study in the early 90s, you know, and the first thing we did was to put all the data in a little diskette and send the diskette to everybody who was asking for it. And people thought that we were crazy, but, you know, in Incanti, that was done on the string of a shoe, probably 500 papers over the last 20 years. And one of the issues is, you know, what means that at some point in time, somebody has, and I don't know how to use this, okay. Somebody has, you know, in age, in biomarker age that is older than chronological age. It could be that they were following the normal trajectory, but at some point something happened to them and they became older. And then for some reason, they can catch up to normal trajectory. Or some other individual were born already older. And so when we catch them, because of cross-sectional data, we see them dead. Or the biomarkers are exactly what we want. They fluctuate up and down depending on our health status event that occur with life. And because of some longitudinal data that are available in the Chianti study, we were able to address this question, at least in part. We will do a lot better in the BLSA, where we are studying 6,500 samples. But if you look at in the Chianti study, and you look at compare the predictive of mortality, the prediction of mortality using only cross-sectional data, only longitudinal data, or using both cross-sectional and longitudinal data, you see that the combination of longitudinal rate of decline or increase and the cross-sectional both contribute to the prediction of mortality, suggesting that it's not happening early in life, but there are modifications over life that really adapt and revealed by this change in the biomarker. And I think that that should be one of the standards that we need to study, change over time. If we don't look at change over time, we don't know whether the biomarker that we're using are useful in any way. The other thing is, why do they do that? I love this picture. Those are fish that start running around. We don't know why. And so why the epigenetic clock does what it does? So in order to address that, we did something that's similar to what Eric did. We look at different type of cell that were circulating. I'll make a long story short, but we measure methylation in this single cell type. And when we did that, we found that those, when you look at a specific cell type, those methylations that are associated with aging are really localized at level of chromatin that look at an answer and promoters. So they seem to have some biological effect. But if you look at the transcription factor binding site, they're co-localized with those age-related differential methylation, you find that the two transcription factors that are most important are the two transcription factors that mediate the hypoxia response mechanism. And so it will make a lot of sense that epigenetic adapt to stress response in terms of hypoxia, because that's what's happening in utero, for example. What is the most important phenomenon that is interact with development? Is lack of oxygen and lack of nutrient. And so that the organisms have a mechanism that adapt transcription to the lack of oxygen and nutrient become more important. And we're studying this in mice, putting mice in alternating hypoxia, and I can tell you that when you do that, you see beautiful changes in the epigenetic clock. I don't have the final result yet, but possibly in the next meeting. How do we do that? How do we study the biomarker of aging? I think that our idea that we could measure the biomarker of aging in human is very, very difficult. If you try to do that, as we have tried, you go crazy, because each single cell type have a different type of autophagy and mitophagy, a different type of DNA repair. But if we could capture some of them in a small population and then capture that signal through biomarker, maybe we should be able to have proxy measures of the biomarkers of aging. I have little time, but I'll tell you that one of the biomarkers of aging that I really love is mitochondrial function. We study mitochondrial function through P31 spectroscopy with MRI, and we found that that mitochondrial function is associated with walking speed, one of the most important functional measures of aging, through the effect of muscle strength, which is our mediator, is a phenotype, and then is also associated with dementia, with accumulation of beta amyloid. So I think that the signaling from energy is probably going to be one of the most important things that we need to start connecting with the biology of aging. Final, this is something that I am been working on with Dan Belsky about, how do we use this clinically? I want to say one thing important. We need to talk more to clinicians. I don't know how many clinicians are here. If we're not talking to clinicians, we will not do translation. And many of the clinicians out there do not believe in what is discussed in this room. And unless we involve them, we will not be able to translate this concept. But one thing that, as a physician, I can tell you that the biomarker of aging probably going to be extremely important in young middle age. When you get to really old age, the functional status of individuals, the walking speed, the memory, the cognition are so powerful predictor of decline that they will overcome any predictive power of any biomarker that you can think of. So we need to start thinking about different biomarkers at different time. And this is all. Thank you very much.
Actually, we do have 1 minute. So if there is any urgent question, you can take it. Anybody? Okay, go ahead.
Is it possible for us to have your slides available somewhere?
Yeah, I think so.
How?
Just send me an email and I'll send them to you.
Okay. What's your email?
You'll find it. Go to
Yeah,
NIA and you'll find me.
Easy to find. Okay, thank you very much, Luigi. So let's thank again, Luigi.
How are you?
Our next speaker is Lee Hood, and he's one of the most famous scientists. He coined the term systems biology, and aging, of course, a systemic process. So we're really looking forward to your insights. Thank you for coming.
What I'd like to talk about is the way of thinking about time. I would say I agree very much with Mike Snyder. I'm all about actionable possibilities that can change people's health. And that's the context in which I'd like. Is it possible to
The first slide. It's the last slide. We need the first slide.
How do we get to this thought? So what I'm going to talk about are
next time I'll let you to hold this.
Study of 5,000 people over 4 years. Both,
okay, do you want to hold this maybe?
And we'll talk about two approaches.
Oh, good. Oh, you can take care of that.
Very early when they're simple, then I would argue this is going to have impact on the aging process. And we call this individual population health.
He's fixing it.
The whole idea is you make a lot of measurements on a lot of people longitudinally over a period of time. And I think a really fundamental point is more and more biomarkers aren't going to be single entities in the future. They're going to be complex compilations of signals that come from the blood or wherever else you're going to assay things that are integrated together to give you higher level functionalities. And we'll see some examples of this as we start. So the genome, as you know, is essentially the source code for giving us the potential we have to develop. And the phenome is essentially how you appear at each stage of your development. And your phenome is how you appear at each stage of your development. And what I think is really important is, in order to take advantage of the things I'm going to talk about, Phenome Health, a nonprofit I started, is pushing the idea of a second genome project where we'll do 1 million people over a 10-year period with whole genome sequencing and deep phenome analyses. And of course, what determines what your phenome looks like as you change are really 3 things. It's 1, your genome. It's 2, your behavior, diet, exercise, and so forth. And it's 3, your environment. All of those are incredibly important. And the assays that we use to assess them are, for the most part now, blood analytes, clinical chemistries, proteins, lipids, metabolites, and so forth, as well as digital health for brain and for body, and electronic health records, and all of those usual things. And it's the integration of these data together using the principles of systems biology that give us deep and fundamental insights. My view is that blood is really a window into health and disease. It bathes all organs, and these organs in turn deposit informational molecules into the blood. And if you can interpret them, you can assess the nature of the organs. And we have organ-specific proteins now for more than 25 different human organs where we can look specifically at the state of these organs. And of course, this data gets converted into digital information. And through the wonder of systems biology, we can map that information into dynamical biological networks that underlie both normal physiology and disease physiology. And of course, what's really changed in the last 5 years is AI. We have the ability not only to use machine learning to get statistical correlations, but we can use knowledge graphs to correlate with all of the data that's present in PubMed. We can use digital twins to be able to make synthetic data and predictions about what's going to happen. And of course, ultimately, the large language models give us the ability, if we can convert digital data into language data, to be able to do unparalleled analyses of how the phenome leads to actionable possibilities that can improve health. The project that we talked about that went from 2015 to 2019, AirVale with 5,000 people analyzed phenomically every 2 years and so forth, has led to more than 30 papers in the literature. And it's led to a series of stories that I'll tell you about in the context. So 1, the early detection of chronic disease. And this came about as a result of the fact with these 5,000 people, 167 transitioned from wellness to many different chronic diseases, 35 of them cancer. And we actually ended up examining first 1 and then 10 more that transitioned to cancer. And what we did operationally was to look at bloods every 6 months prior to the clinical diagnosis of the disease. And what we were able to show beautifully is in a person that we called Eve, that there were 5 proteins that appeared at the earliest stage of her blood analytes at very low levels, and then increased as you move closer to clinical diagnosis. And those 5 proteins mapped into 3 major networks that were found in stage 4 pancreatic cancer, her particular kind of diagnosis and all. Okay. So we looked then at 10 individuals that had cancer, and in all cases found exactly the same kind of thing anywhere from 1 to 4 years prior to the onset of disease, we looked at one type of cancer that had 3 different transitions, and we showed it had common markers and distinguishing markers, presumably that define the different subtypes of prostatic cancer. And of course, with this million-person project, we estimate we'd have 200,000 or so of these transitions, and that would give us the data we need to precisely be able to predict at the first instance of a elevated series of proteins what in fact is going to be the chronic disease that will be later diagnosed. And of course, this is really, as we see it, one real key to prevention of chronic diseases. If we can have these markers, and if we have people on a regular 6-month trial basis, we have the ability to predict disease well before it becomes clinically manifest, and to think about how we can reverse that actual disease. And the other thing that we did that was absolutely revelatory is in Eve, we examined 9 different disease-perturbed networks that were common to chronic pancreatic cancer. And we were able to show, as we moved in the blood draws closer and closer to the diagnosis, basically an exponential increase in the number of biological networks that had become disease perturbed. And that argues compellingly that we should reverse all chronic diseases at their earliest instance of detection when the disease is relatively simple and so forth. So the second story is really an intriguing observation where we can use polygenic scores that will translate into individual genetic risk and correlate these with blood analytes in interesting ways. And we did this in a paper that appeared in PNAS back in 2020. And essentially what we did was plot on the x-axis increasing genetic risk, and on the y-axis concentration of each of 800 different blood analytes that we tested against 54 diseases. We found that more than 800 of these correlated in a really interesting way with increasing genetic risk, that is, increasing and or decreasing. And what we found that was really striking is when we looked with the axis again, increasing genetic risk on the x-axis, but on the y-axis, clinical diagnosis, what we could see is in the upper 5% of the people, the clinical features place them very close to people that had transitioned over to the disease. And exactly what the same was true of the analytes that we were able to correlate with these different things. So it means for the 250 diseases that we can make genetic correlations with now, we can actually detect the 5% that are very close to transition, and again, use them to reverse the disease. The third story had to do with healthy aging and health span, and it basically revolved around the fact that we had people spanning from 21 to 93. We divided them into 10-year categories, and we were able to show beautifully, as you aged, your ability to control the expression of the 3 classes of analytes diminished in quite a striking fashion. And there were people who migrated up on the upper end with extreme divergences, and people who migrated on the lower end that controlled it much, much better. In a sense, these are the different aspects of aging. And what we were able to do is develop an algorithm, and we could use any one of the 3 blood analytes we looked at, clinical chemistries, which were great because they give you actionable possibilities. Metabolites do give you some actionable possibilities as well, and proteins. All 3 of them actually could be used to determine one's biological age, the age your body says you are, as opposed to what your birthday says. And what was striking is when we examined the 5,000 people in the Airfield program, we could show for every year a woman stayed in the program, she lost 1.5 years of biological age. So on average, they got 6 years younger by this aging metric we have here, and men were 0.8 years per year. I had a friend who lost 10 years in that 4-year period, and my biological age right now is 15 years younger than my chronologic age. So the question of how much this can be perturbed, I think, is absolutely a fascinating question. But what is really clear is that you can decrease the rate at which one ages biologically and so forth. And finally, I would say, if we are able to study, well, let me just say, we studied 40 different diseases in the Airvale population. In every single case, the biological age was increased and level and so forth. So we think this is a very powerful metric for being able to
no time.
No time left.
We need to start.
So let me just close up by saying scientific wellness and these two approaches we've taken to early detection of chronic diseases, I think, are interesting biomarkers that give us hope that we can really profoundly change the rate at which we age. Thank you. Yeah. Shall I give you this? What?
You're from Harvard and Stanford.
Oh. Okay, okay.
Hello, everyone, and welcome again to the first Biomarkers of Aging Symposium. My name is Mahdi Moqri, and for the next 10 minutes, I want to tell you more about our other activities besides helping with this symposium. So many of you are using biomarkers in either population studies, basic research for drug discovery, or clinical studies. But regardless of the application, there are these 4 challenges that we need to address in all of them. Which biomarker to use, characterizing different biomarkers, do they work, can we validate them, and how to implement them in that specific setting, in the clinical setting or drug discovery setting, and how does your biomarker compare with other biomarkers? How do we benchmark them? And we've been dealing with the same issues in the labs that we've been working at Harvard and Stanford for a few years. And we thought it's the right time to put together the community and address some of these challenges together, because some of them cannot be resolved within one lab. And 3 of my mentors kindly agreed to join this core committee, and followed by our Eric Verdin and Andrea Meyer. Great complementary expertise from the clinical and translational domain. And these are the most collaborative and pleasant people to work with. I'm glad we have them. We also invited an advisory committee to help us this year. And we are also expanding this committee. And we also invited 30 other experts from basic research, population study, all the way to clinical studies, and people from United Nations. People who are translating this for the larger population for healthy longevity. And together, we put together a roadmap. We thought these are the activities that we can help with for characterizing, validating, translating, and benchmarking. So the main activities are a set of manuscript consensus building manuscripts toward these goals, and our annual events, symposiums, and an annual challenge that benchmark these biomarkers. It's the first time we're trying. We might fail, but better try than see the result. For our consensus manuscripts, we put together a Delphi-based process with a core drafting team. Thanks very much to Chiara and Jesse with drafting all of our manuscripts. And then we invited all these experts for the content, and back and forth with these experts for missing contents, for revising the contents, and together with collaborative writing, driving consensus as much as possible to have a working solution, because we have a practical problem to solve. So balancing between intellectual disagreement and making some working consensus so we can get some stuff done. And with the improved process, we had Luigi, Ferrucci, and Steve also joining on the leading, corresponding with Vadim, Gladyshev. And now there is a team of, if there is any remaining conflict, we need to publish these papers. We have a good team to take care of that. So for the first work, we spent around 6 months with this group of around 25 different research groups. And out of those 6 months, the first 3 months, the hardest part was having an agreement on the definition of aging and biomarkers of aging. And it was just surprising how many different theories there exist. There are more than 300 different theories of aging, and every one of them proposes a definition of aging. So for us, we have spent 3 months meeting with individual one-on-one with all these experts, and then using collaborative writing to drive some definition, consensus working definition for aging, for biomarkers of aging. And after that, we were able to extend the FDA biomarkers classification framework to make it appropriate for biomarkers of aging. And we see that omics, different kind of omics, especially epigenomics, are becoming more and more popular in the field. And a lot of these current biomarkers are predictive, especially they are predictive for mortality. But we know from the broader field of biomarkers, we need response biomarkers for clinical interventions. We need discovery biomarkers. And for us, it's how can we get there with the help of this community. And the co-authors of this work, many of them are presenting today. I want to thank in particular people who spend more time with us with this, Jamie Justice, Alan Cohen, Dan Belsky. And I remember it was a collaborative writing platform, but at some point, Alan and Jamie was using the comment function to chit chat. It was a fun experiment. And for the follow-up work, and the result is now published. Oh, there is no back button. All right. The result is published a few months ago in the Cell. It's available, and we are encouraging everyone to use the consensus definition used established in this work. And for the follow-up work, we invited more people with population expertise and with validation expertise to help us to see how we can validate these existing biomarkers. There are already a good number of biomarkers. We have 50 epigenetic biomarkers. A lot of them are for tracking aging, and some of them are better validated, some of them less. So we asked the community how we can start building a framework for validating them in different settings. So the roadmap that we propose together is, instead of focusing on validating one biomarker in one population, we need to move to validating multiple biomarkers, comparing and testing multiple biomarkers in multiple diverse populations. And that's the key, to have these large cohorts, they're breaking the walls, getting more diverse population data so we can validate our biomarkers across them. Different ages, different background, different settings, they're more diverse, they're more likely that they become translational. And we also made some interesting observations that some of these biomarkers are now working on a large, these are validation data that are being validated in large populations and very diverse cohorts. We have one that's now using 26 different cohorts. And some of them are reporting very, very interesting and promising performances One of them is reporting if you use it for population study stratification, the two groups that you get, one of them have 10-fold chance risk of death compared to the other one. So these are amazing number that have very real clinical implications. And we also see that other independent groups have started to compare these biomarkers. And we are getting toward establishing some sense of comparison, some sense of benchmarking, especially for the epigenetic ones. And now multiple different biomarkers from different groups have been tested in the same cohort, and we can see how they compare. Still a lot to do, but I think it's a good start. We also identified some of the less utilized cohorts that are very promising, very rich data sets that are already out there, but they are not as known, not as common, some of them, as the most commonly used cohorts for biomarkers. Together, again, with the group, we put a vote, very transparent, very fair system. Everyone suggested their own favorite cohorts, and together we identified some of these less utilized cohorts that we encourage people to use for validating more biomarkers. We also have some additional contributors for this work presenting today and available today. And I'm highlighting here more people from population expertise that were invited to add to contribute to this work. And this work is also forthcoming in Nature Medicine. So I think it's just having all these experts help us to stay on the right track. And on the translational side, we leverage this event. And a few months ago, we started distributing surveys to our participants. So everyone who is giving a talk or on a panel or even moderating, all of them have received an invitation to respond to this survey about what's the best path to translation of these biomarkers. Similar discussions that we had with our consortium panel, but every other speaker and panelist and moderator have been invited to participate in this. So far, we got 29 response, but preliminary results show that these different criteria, they matter, especially for your context of use. If you are doing predictive or diagnostic biomarkers, you might care more about a diverse population and less care about response to geroprotectors. But if you are building response biomarkers for testing anti-aging drugs, you really care about response to these geroprotectors. So it's good to also see some convergence of opinions over there. And we also have some exciting news, exciting announcement. So make sure you don't miss the 5:30 talk by Jesse and Dane, and our closing remarks by Vadim. We have some good news for some plans and some good news and some more announcement regarding to benchmarking and a competition as a challenge. I don't want to spoil it. Yeah, finally, I want to thank the labs that I've been working with for the past few years. I started at the lab of Vittorio Sebastiano. Thanks for accepting me. I was completely in a different field. So thanks for letting me into the field. And Mike Snyder, I worked with in his lab for 2 years. And Vadim Gladyshev, I'm currently spending most of my time at the Gladyshev lab, one of the best place to be with Jesse and Ludger and everyone else, and Albert. Thank you very much. And I want to have time for questions if possible.
We do have a couple of minutes for questions. So anybody, any questions or comments? Yeah.
Thank you for a great talk. With regards to industry participation, and in particular clinical trials, what is it going to take to convince big pharma to utilize biological clocks or aging biomarkers when interpreting results of clinical trials, whether in a disease or a therapeutic area that is related to aging or not? I mean, we know now, you know, that patients are grouped by age, and that's chronological age. But do you think, can you comment on the potential of utilizing biological age to correct for chronological age when interpreting clinical trial data?
Yeah.
And what is it going to take?
Yeah, So the good thing is we also have an industry panel today, and we already have a few CEOs from those related industries, and they will be discussing that for 45 minutes. But in short, we already see some promising results from the industry. Just to name a few, Gero is working on these translational problems. BioAge is working on that. In silico medicine is working on that, and we have representatives from all of them that will discuss that in details.
Okay, another question here.
Just a follow-up question. My name is Federico Gutset from Symbiont. The question had to do with what it would take for the industry to take them up. From a diagnostic perspective, there may be a relatively fast path forward at some point. From a therapeutic perspective, there are a number of requirements that we need to define for the biomarkers that are going to be used that are no-brainers for therapeutic product development. One of them, of course, is reversibility. It's okay going forward, but what happens when you provide a therapeutic agent that is supposed to reverse it? Can you measure that with those biomarkers? They must be reversible. The second part of the story is that an early conversation with the FDA is really required, whether or not you want to define aging as a disease, because these biomarkers could be the key to define how we will have therapeutic agents in the future. And for that purpose, you need to make the agency aware of where these biomarkers lead us, okay?
Yeah, I agree with your comments. Very good point. And we have started with having this in mind, that at the end we want to talk to FDA and regulatory. And the reason that we started with consensus is FDA requires at least traditionally scientific consensus about the context.
No.
Like traditionally.
No. It requires that consensus in being able to qualify the biomarkers for everything at every time. In order to have these biomarkers used in clinical trials, as surrogates, for example, we need to have a type C meeting, provided that you have enough data to justify that surrogate. But if you wait for the community to have this, you have 16 Egyptian dynasties.
Very good point, and I like to hear that point.
Yeah. Yeah, thank you. And then I think it's a good time to discuss it during the coffee break. Just one small announcement for the poster presenters. Please put your posters on, because the posters, it's already start. We'll look for them. Thank you. Now coffee break. See you in 30 minutes.
Thank you. First session is done. Oh, I missed one. Thank you.
So let's see.
We can also help you in the room if it's.
Yeah, I can.
Wonderful. Thank you.
Yeah, we haven't talked about genetic risk. You want us to take our badges off? Or?
Also very much so grant and foundation funded, so it looks like kind of and then
Good. Good.
we've been waiting for this moment to take a photo.
How about that? Well, this consortium,
Yes, there's a first.
Here I just wanted to hit the three panelists submitted too, but if this one could stay up while Verdin does the introduction.
Yeah.
that would be good, 'cause it needs the names.
Oh, so
And then there
let's-
should be another.
Like this one?
Uh,
This one.
Yeah, Geroscience Panel, that's right.
Okay.
Yeah. So
Um,
Eric introduces with this, and then we can shift to that. And then at the end,
Okay.
I don't know actually if we should just leave the last one of this or go back to this one.
Yeah, oh, yeah. So the screen's frozen. So for this until we
Eric is
start.
done, yeah. And then when they start the panel, we
I'll
can.
unfreeze it and I'll start showing this.
Perfect.
And then I guess
Lou. Yeah.
Lou's, he can use this
Okay.
to advance.
Okay, great. We'll let him know.
Cool.
Okay, thank you so
Yeah,
much.
you're welcome.
I can put one away.
So this is the advancer. So it's on the slides right now, but the screen is frozen. So once the panel begins, I'm gonna unfreeze the screen and then you'll start seeing that. And then when you press the button,
You'll bring that up.
yeah, you can actually see it there now.
Can I try
Yeah,
it now?
yeah, go for it.
Oh, I see. So whose is this?
I don't know.
This looks like the last one, actually.
This is the one I was asked to play or have up for the panel. And I think
I don't know who's who, and each one had one slide, but it looks like we've got, oh. Huh, I don't know who's who.
I think it's just for
Oh,
the
Yukon
whole-
would be George, okay. But he's the last one. So,
I think they're all Yukon.
oh, yeah, they are all Yukons. That doesn't
I
work.
don't know how that happened.
No, those are all George.
I think that if it might be good to ask Chiara about the slides or Lewis if you
Yeah,
sorry, yeah, if you have a question, because I'm not so sure about it, but I was just asked to give you that.
So I'm not sure that they all have slides now that I see this.
Yeah, I think it's just like for the whole panel.
Yeah, I was told that each one had one slide. I don't think that's true.
Okay.
So let me go find out.
Okay.
I'll do it. So here says resilience.
Yeah, that's the only one. I don't know what
Okay.
the other two are.
Well, hang on.
That's
What's
not
this
mine.
one? So that says Yukon.
That's wrong. That's
Whose is this?
I think that may be Vera.
Well,
And
that's
and the next
biology.
one, and that's
That's me.
Yeah, so that's
Oh, why
Steve.
did you put Yukon at the bottom? I'm not Yukon.
That's right. That's the
Why did they
problem.
put Yukon at the
And,
bottom?
it's frozen on the screen now.
No,
No, no, no. That's
No.
not the issue. The issue
Can
is
you-
that all three
mislabeled.
slides have Yukon on the bottom.
They put the content onto my slide, onto my template.
Yukon.
Can you, is there a way of deleting
Just delete
Yukon?
that. Yeah.
I don't know why.
I
I'm
had
trying to.
nothing- I had nothing to do
No,
with
that
it.
one has, that one does not have Yukon. That one does not have Yukon. That's great. That one does.
welcome everyone. We're going to start our second session, which is going to be comprised of two of these roundtables that you are now familiar with. The first one is going to be handled by Louis Lipsitz from Hebrew Senior Life. And the second one will come with Neil Barzilai, who's going to talk about the NIA biomarker initiative. So Louis, you have the floor. Good.
Thank you, Eric. Welcome, everyone. It's really my pleasure to be the moderator for this particular session. As you heard, I'm Lou Lipsitz. I am a geriatrician taking care of older people and a clinical investigator at Hebrew Senior Life's Institute for Aging Research and at Harvard Medical School. As you heard, we have three presenters in the area of geroscience, and we're going to begin with a brief statement from each of them about their particular, who they are and their particular focus on the field of geroscience. So we're going to have one slide for each, and we'll start with Dr. George Cashel from the University of Connecticut. And you want to use the clicker, George?
Okay, thank you. So I too would have changed the slides had I seen all the wonderful talks earlier. So I'm George Cashel. I'm also a geriatrician. I'm a physician scientist at the University of Connecticut, where I direct the UConn Center on Aging, our Pepper Center, and also our Senate Tissue Mapping Center. So I was asked to, and I do a lot of research in the geroscience space. I'm an MPI of the Translational Geroscience Network. And I was asked to comment in the context of biomarkers and resilience. And so I'm going to have to turn around. Otherwise,
you can see them.
So first of all, not all markers of aging are meaningful from a geroscience perspective. And I think we've heard a lot of that earlier. And I'm just going to repeat a little bit what was said earlier, that for a biomarker to be useful in terms of gerotherapeutic testing, which I don't think it's quite the same as a biomarker of aging, they're related, obviously. It needs to be reliable and reproducible. You need reliable and reproducible assays to be feasible in context of a proposed study, which can be really challenging, particularly if you're doing a clinical trial, particularly if you're doing a multi-site clinical trial. It has to be relevant to biological aging. It has to consistently be able to predict all-cause mortality, clinical and functional outcomes. You'll hear more about that later. And there needs to be evidence of responsiveness to intervention, the particular intervention being tested. That's kind of the minimum. Now, there are problems with this approach, and one of them is that in many situations, there is a place for preventative strategies, geroscience-guided strategies, long before there's evidence of altered physiology or even biomarker detection. So we've heard a lot about that earlier today. And the other issue is that gerotherapeutic trials could last years or even decades, depending on what outcomes you're measuring. If the outcome is mortality, it could certainly be very long. So the idea of resilience, and many of you do work in this field, is that in the context of biomarkers of aging and biomarkers of gerotherapeutics, is that if we study resilience, you have to assess responses to stressors, including temporal return back to homeostasis. So that gets back to the issue of having of multiple measurements involving both the biomarker as well as the related physiological measures that have to be done repeatedly. And I think that the reason why this may be really helpful is that clinically, we certainly see many situations in which if you see a patient, and to give you one example, you simply, you measure their blood pressure sitting, you're going to find a lot fewer abnormalities as opposed to measuring their blood pressure as they stand up or as they exercise. Another example is glucose tolerance versus diabetes mellitus. If you simply measure fasting glucose, you will not pick up as many individuals with dysregulation of glucose metabolism as you will if you challenge them with eating with a glucose meal or a Krispy Kreme donut. And so our group does a lot of work in terms of, as you heard earlier from Eric Verdin, does a lot of work in immunology of aging, particularly in terms of vaccinations. And so I'd like to propose that vaccination in many ways is a very appealing paradigm because it's both a test of resilience, where you actually introducing to the individual a foreign antigen. And it's also a way of providing a gerotherapeutic boost of immune resilience in that context. And therefore, if we're looking at resilience, a biological basis of resilience and responses of biomarkers of aging to these stressors, you're talking about a time frame that lasts from weeks to months as opposed to years or decades, and may have some. And actually, the NIA has funded major initiatives, both in terms of studying resilience in mouse models. Some of you here were involved in that, as well as funding two large human trials that have been delayed by COVID, unfortunately, but will be coming up in the next year or two. Will be coming up with very important information about resilience in the context of stressors such as knee replacement or hip fracture or others. Okay, I'll stop there.
Great. Well, why don't we turn the microphone and the clicker over to Vera to talk to us about her focus on biology.
I'm Vera Gorbunova. I'm at the University of Rochester in upstate New York, and I direct Aging Research Center there. So my research is focused on aging, of course, and it includes comparative biology of aging, where we study long-lived animals from whales to naked mole rats and bats. And the goal is to understand what makes them so long-lived, and then to ultimately transfer these mechanisms into clinical practice to benefit humans. And we also study genome stability. So in relation to this meeting, two topics that we are working on and I'm particularly interested in are biologically relevant methylation clocks. And Steve already mentioned, and other speakers as well, that it's very important to find functional component of the clock, because some of it may be just accumulation of noise, completely stochastic, and each of these CPG sites may by itself not carry particular biological information, but at the same time, within within all this soup of CPGs, there may be some that are meaningful. And we are particularly interested right now in exploring transposable elements, because we discovered that when transposable elements become derepressed with age, they can drive inflammation, which is a systemic response, and it can be underlying cause of multiple age-related conditions. So we are really interested in building transposon-based clocks. One limitation to that is that the Infinium array that was mentioned is actually pretty much depleted of transposable elements. So we are using for other alternative platforms that would allow us to do that. So this is one topic. I have a little cartoon there on the slide showing human chromatin in old individual where transposons become activated as genomic parasites. And you see all these transposons are active, and now they can start driving inflammation. And they may be actually the basis for functional clock. So the second topic we are very interested in is why, in comparative sense, so why long-lived species have slower epigenetic drift. And I included a figure that will come out in Nature Aging in News and Views for 2 articles that discover these scaling laws, that long-lived species have slower rate of drift of epigenetic age. And I find this absolutely fascinating, and it raises questions as to why that is. So what are the mechanisms that slow the drift in long-lived animals? And if we could find these mechanisms, and well, we found some of them, so we work on some epigenetic enzymes, but if we really get complete picture, so then we could target those enzymes, activate or repress them depending on what they do. And this may be the way to actually target underlying causes of aging. Well, if indeed changes in methylation, some of them are causal. So these are perhaps 2 topics I wanted to cover.
Great, thank you. And Steve?
Yeah, by the way, thanks for inviting me. That's really a terrific meeting so far. And I don't think I'll need that. Oh,
you need it for your
slide. Change the slide.
Here.
How's that?
That's it.
Okay. Thanks. So I'm a doctor. I practiced internal medicine for 30 years, also an epidemiologist. I lead a coordinating center that has really large cohort studies, prospective cohort studies with patients drawn, for example, from Wake Forest, where Steve Horvath is the lead. And that has yielded about 30,000 individuals followed long-term with data, specimens available. All of that's available on, almost all of it's available on websites. You can download it. We can talk later about what, what access means in that context. So that, and our coordinator center also runs multi-center clinical trials, trials that have established by reducing the risk of fracture, essentially all the drugs that are now used in clinical practice and approved by the FDA. We'll use that data along with other trial data right now to develop an FDA-approved surrogate marker. That is change in bone density, which we believe will become a surrogate marker that allows trials to be less than 15,000 for 4 years and maybe 300 for 2 years. So we have a lot of experience in what's entailed with a validated surrogate marker. But I want to pose an issue, and that is on this slide, good predictors, strong predictors of aging-related endpoints that turn out to be poor or lousy surrogates. One of the most recent and my favorite is vitamin D levels. How many here has had vitamin D level measured? Woo!
Wow.
See, this being almost universal because of the consensus that vitamin D is a strong predictor, as it is, of mortality and many other aging-related outcomes. And you can change it by giving vitamin D. You can increase it back to a normal range. So that's very, that's the kind of strategy we're looking for here for a surrogate marker. And guess what? Large randomized trials, we're now about 40,000 individuals, have found exactly zero effect of of doses of vitamin D on any meaningful clinical outcome, mortality included, even in those whose vitamin D levels are low and are then normalized. Doesn't work. And it's a good example of a really terrific predictor that turns out to be, as far as I'm concerned, useless surrogate marker. And then the last, one other thing it's important about that is we got to think about safety, because we got drugs, potential drugs that interfere with a lot of aging biology, and maybe they also have adverse effects. Got a couple of examples to come back to, but in the case of vitamin D, doses above 2,000, 4,000, which you can get from your drugstore, 10,000, which you can get from your drugstore, cause harm, faster bone loss, muscle weakness, and an increased risk of falls. And now I'll point out the amyloid antibody trials, which many of you have heard about because they're controversial. These, we know that amyloid is strongly associated with dementia, with Alzheimer's disease, right? And therefore it's a great target. And so there have been anti-amyloid antibodies that have been tested by companies showing that you really do a great job of clearing amyloid and you have little or no effect on cognitive function. And I think that these are sobering and should leave us both humble and also rigorous in what we end up trying to do as evidence for primary care practice. There are many more examples we can talk about later on. That's it.
All right, why don't you hand me the clicker? Let me take the clicker from here.
Ooh.
All right. Well, thank you for those perspectives. It's interesting. We've heard that some of you have favorite biomarkers. Some of you have critiques of existing biomarkers. And one of the things that we've heard a bit about so far, I think Luigi and Eric both emphasized this, is the dynamic nature of biomarkers. In fact, all of you have mentioned that. They're dynamic, they're nonlinear. They may change over time. And as in the case of vitamin D, may be toxic if too high and toxic if too low. So how do we deal with those dynamics? One of the things that has become clear to me in my own studies, I'm doing a clinical trial of 2 senolytic agents for their effects on cognition and mobility and a variety of SASPs or senescence biomarkers. And as we recruit subjects, we follow them carefully, we give them their medications, we measure bloods along the way, and all of a sudden one of them gets a flu vaccine or gets a COVID vaccine, and the biomarkers, the inflammatory markers I'm measuring are probably all thrown off. Another one gets a dog bite and gets put on antibiotics and develops hepatitis as a result of the antibiotic. Again, those biomarkers are probably not going to be valid as markers of the cognition, mobility, and senescent cells that I'm interested in. So how do we deal with those dynamic nonlinear measures? George mentioned provocative tests, which I think is one approach. So why don't I start with you, George, and we'll go around and talk about how each of you might deal with that. Are you interested in more of the static or dynamic markers, and if so, how?
Well, I think first of all, in terms of just good practice, good research practice, you try to standardize things in terms of times of blood draw and fasting, et cetera. I mean, just eating, for example, has effects on biomarkers such as IGF-1, IL-6, and others. So I think, and they have many circadian relationships as well. So I think first one approach is just to standardize things so you avoid that kind of noise. And the second is that if you're able to do more frequent sampling, and there's some very innovative methods of doing that, is to look at those relationships over time, and it actually provides an opportunity to look at issues like resilience, for example, that otherwise would not be possible.
Vera.
Well, I think absolutely we need a good level of reproducibility. Otherwise, when there is too much noise, it's very difficult. I think initially this was the reason why methylation clock became so popular, because it was more reproducible than other types of omics. But I think now there are newer clocks being developed that combine within them various types of omics data. So maybe that's really where science is going, to have a combined score.
So I thank Luigi for some of the early observation that cross-sectional associations of some marker with age look stronger than, well, excuse me, that when you do it longitudinally, what looks like a lesser effect of age on these markers turns out to be steeper, because, for example, some of the reasons are older people are less likely to volunteer. They're the ones who have poor levels, and therefore you're misled by the cross-sectional data. And so it's really important, and Luigi can speak eloquently about the importance of that longitudinal data. And it also, longitudinal data are stronger in terms of making causal inferences. There are a lot of reasons confounding why cross-sectional data is not as strong. Longitudinal data also is important to help plan clinical trials. I mean, if we're going to plan clinical trials, what we really need from our cohort studies is how the things we're interested in change over time in various populations. There's a lot more to be said about that, but longitudinal data is important for at least those.
And actually, I just made so the discrepancy between cross-sectional and longitudinal data is not something that we learned yesterday. There is actually a series of seminal papers published by Nathan Schock, who everybody's heard of, going back to the 1970s, showing that in terms of kidney function, measuring creatinine levels, you get very different results if you do cross-sectional versus longitudinal analyses. And while averages tend to decrease in terms of renal function, there are some individual that, you know, you have heterogeneity in terms of patterns, where some people decline, others don't.
Yeah, thank you. I think longitudinal data are really critically important. So this particular session addresses biomarkers for geroscience. So geroscience intends to identify fundamental modifiable mechanisms of aging that may become targets for future interventions to prevent age-related disease and disability. So I'm curious to hear what, I see three clinicians here, George, Steve, myself, who see patients. And I'm curious as to what your favorite use of biomarkers might be. One use I've heard over and over again is to predict mortality or longevity, perhaps. But as clinicians and perhaps people in geroscience interested in addressing those fundamental mechanisms that lead people to disease and disability, mortality, though it's appealing because it's a fixed endpoint, you can't miss it, may not be the right reason to identify biomarkers, particularly in the clinical sciences or in geroscience. So, George, what's your favorite use of a biomarker? What would you suggest we should be
clinically or in terms of research? Or
let's do clinical, but we were trying to help human beings.
Well, I mean, first of all, if it's in the context of what matters to our patients, so I think what's really important is that we haven't asked our patients what's important to them. And I think a really important development in geroscience is that after the last geroscience at NIH, AARP is very much on board. And they've actually agreed to do something which I think has to be done. And it's actually to use their expertise in what we know nothing about, which is marketing and also getting public opinions, find out what actually matters to people out there from a geroscience perspective, because everything we've said so far is based on what we as individuals feel. So I think that's the first issue. And I think the other issue is that although I fervently believe in the geroscience concept, I do think that when it comes to biomarkers of geroscience, I think there are three problems, and they're all disorders of thinking. I think there's a lot of circular thinking where biological aging predicts a biomarker, and therefore biomarkers define, biological aging defines biomarker, biomarker defines biological aging. And there's a lot of visual and magical thinking as well. I think that a lot of, I think this is a great step forward in terms of actually doing the rigorous work of validating these biomarkers in the context of real populations. Otherwise, it's just thinking.
And ultimately, we want to validate them in terms of an individual patient.
And of course, I realize I haven't answered your question.
Right. That's all right.
Well, for me, I guess the most straightforward application would be in clinical trials, because I think doing human lifespan trials wouldn't even be possible without the biomarkers. We cannot just wait so long. So it would be fantastic to have a way to evaluate interventions on relatively short term. If we can do 6 months of an intervention, like a supplement, and then measure the biomarker. So this is one, and that's probably the most impactful. But then I also want to talk briefly about another way, which has to do with functional biomarkers, because that may be something that we can target. I want also to give a little plug to Steve Horvath's preprint, where we collaborated and built a different clock that actually measures not chronological or biological lifespan, but it looks at maximum lifespan. So apparently species, you can predict from methylation patterns maximum lifespan. And it's not the same. It's not the same set of CpG. It's completely different. It's very difficult to change them. Like if you're human, you have this signature. If you're a dog or a mouse, you have a different one. And I'm super excited about that, because if we find an intervention that changes these CpGs, so then we can actually have potential for massive change in lifespans. People came up with interventions in the mouse. You can make mouse live 10% longer, maybe 20. Nobody came up with intervention to make the mouse live 10-fold longer. But maybe if we can, based on this type of a clock biomarker, maybe we can do that, because it really paves the way.
Yeah.
I'll try to be brief to say 5 things, but Doug Keel's going to talk a little bit later, I think, about the kind of phenotypes as endpoints we want to talk about. And Steve Kuchevsky's doing really nice work that I can't disclose yet, asking patients what really matters to them, because what we choose has got to be salient to the patients. Otherwise, they're not going to be interested in the therapeutics. So I get to tell you my 2 favorite ones. One, a potential, well, SASP is also one I think's got a lot of promise, but I'm not going to talk about that because Nathan will. And the 2 I like, mitochondrial function, which we can also probably measure in blood. Well, promise could be a marker that you could use clinically if we've got therapeutics that improve mitochondrial function. We've got one now. That's aerobic exercise. That's too bad. I can't do it. So many won't. But if we got something better, that's going to be a really powerful measure of an underlying mechanism. And now my favorite, something that everybody in this room already has, in your blood count, and Eric Verdin, where are you? Something that you've got to include in your, Eric, this is something called the red cell distribution width. That is, the size of our red blood cells becomes more heterogeneous as we age. We don't know the underlying process, probably has something to do with the stem cells that give rise to red blood cells. The more variability there is, the worse you are. And so it's in percentages. And we see the strongest associations probably and most consistent across aging outcomes, across fitness, even mitochondrial function, mortality, you name it, it's a strong predictor of things we care about. And it's cheap because everybody's got one here for free. And the last, we've done interesting work in our health system showing that in those records, people with a high RDW are more likely to be readmitted after they're discharged, and they're more likely to die during hospitalization. The CEO of our hospital is very interested in what we can do. It's not causal, it's just predictive, and identifies people for whom we may be able to do something before they're discharged to prevent them from coming back. That is an actionable predictive biomarker, not a surrogate, but I think it's quite actionable.
So we've heard that biomarkers can be predictive. They can be certainly mechanistic. In some sense, they're also just mediators that become targets. If we're going to do longitudinal studies, none of us are going to live long enough to do the definitive longitudinal study across an entire lifespan. So we need to find these surrogates. But are those really surrogates of what? And in our study, where we're looking at the effect of senolytics on cognition and mobility, the biomarkers are really mediators, right? That might be targets in the future for disease. So how do we think about that? And how do you think about that in each of your studies? Are you studying biomarkers for their mechanistic properties, for their preventive, predictive properties, or are you seeing them as really mediators of the development of disease and disability that can be targets for future intervention?
So in the context of testing gerotherapeutics, it's the 4 criteria that I showed on the slide. So it's all of the above. It's all of the above, and I think you need to, you really need to consider all of those. And I think again, it depends on what outcomes you're targeting, and what your intervention is, and has to be custom made. So I think that when people say, what's your recipe for testing a gerotherapeutic, it's kind of a nonsensical question, because it really depends on what the intervention is and what you're targeting. It has to be, I think, has to be custom designed, and it really requires some careful thought.
Well, I agree with that. I think, and that's also the power of AI. You can train the system to whatever outcome you want. You can focus more on functional aspects. You can focus more just on predicting a particular health outcome. So I think we have really a whole field, and ideally you would have different sets of markers that could be applied to patients with different predispositions to give more accurate result.
We do a lot of work with predictive biomarkers and such, but I'm going to spend just a moment with a cartoon of what a surrogate marker requires. So it starts off with something that has to predict the outcome. So it has to be a predictive biomarker. It has to respond to the treatment. Now that certainly makes sense. We have lots of surrogate markers that do that and fail. And when they fail and you look back at it, it's because the mechanism, that is the prediction and the response, had different mechanisms. And people really didn't understand that. And so understanding the mechanism by which the prediction occurs and the mechanism of the response, boy, for osteoporosis, bone density is easy. That's how the drugs work. And that's how the fractures occur. For others like vitamin D, we didn't really understand that biology. And now we're starting to understand that it has a lot to do with the binding protein that binds the vitamin D and can't really be changed by the supplement. So understanding those parts, mechanism, prediction, response, and the mechanism by which they might be knitted together for an outcome.
Yeah, I think that's really a good point about why surrogates don't always work, because we're looking at the different mechanisms of prediction and response. Well, getting at some of your individual work, Vera, you work with different species of animals. It's quite interesting. Some of the longest lived species from what I understand. So how important is the cross-species validation of biomarkers? We heard earlier that we should find biomarkers that sort of apply to all organisms. Is this your belief, or do you think, is there value in doing the cross-species work, or do you think that each species has its own certain cocktail of biomarkers that you might look at?
Well, I think ultimately we need the biomarker that works best for humans. Because this is one species we are most interested from clinical perspective for selfish reasons. Cross-species work is very, fulfilling, fascinating, just because it's interesting, right? But also because we can learn a lot. We can learn from these species that are longer lived, what mechanisms operate there that ensure their longevity. Whether we need a perfect biomarker, for example, for bowhead whale to know how long it lives, probably not necessarily. But if there is a biomarker that we know works similarly, and we find particular mechanisms in the whale, we can then test. For example, we can try to generate a drug that we can give to people and to see whether it will affect biomarkers in the same way. So I would say, I wouldn't call it's not the goal to have a biomarker that works perfectly to all species. And actually, Steve Horvath has developed many such biomarkers, and also a universal biomarker. Universal biomarker does work, but it's not as accurate as individual species biomarkers. But each one has its value. So I think these long-lived species are really good in terms of discovery place. We discover things, but then whatever interesting we find, we bring into human, and that's where we need the most accurate biomarker.
Yeah, thank you. Yeah.
Please. Vera and I are working together on, double-strand DNA breaks in humans, some of our human specimens in muscle, and then will be in blood. And having the cross-species data in which to put that context is really exciting, because she's found that the DNA damage response is so important. Putting the humans on that scale using the specimens we're working on, I think is really going to be helpful and compelling as a result that we hope we're getting.
Well, thank you, Steve. And we should also try to get some whale muscle for you.
That would be hard, I would think. Oh, that's great. Let me pose a question to George. I really appreciate your focus on resilience and response to stress. And after all, that is what we want to achieve in geroscience, is to improve everyone's resilience and ability to overcome or prevent disease in the first place as we get older. That, I think, is really a fundamental goal of geroscience. So the problem with resilience, and with testing it, is that it's situation dependent. So I'm wondering how, in your own research, you might incorporate that concept. You mentioned some of the resilience measures that you might suggest, but perhaps tell us a little bit more about how that might be used.
And also to answer your earlier question, so in terms of what really works clinically, I mean, to me, the most useful marker, predictive marker of how a patient walking into my office is how fast they walk, by far, and that accompanied by how many medications they take, accompanied by how many chronic diseases they have, what we used to call, before EMR, we used to call the congestive chart failure before electronic medical record. And that's really hard to beat, and mobility in particular, because it's also not only so highly predictive, it's also what's the most meaningful to most people in terms of remaining independent. So it's really hard to beat. It's also a test of resilience, if you will, which really, and I think many of us think the reason it's so highly predictive is because it tests the whole body, and you literally, it's kind of a stress test for the entire body, not just muscle, it's heart, brain, everything.
Yeah, I think that's a good point, that we really need these biomarkers that really do reflect the integration of multiple systems, the systems biology, as we've heard, because in fact, EngageSpeed does that by integrating not just muscle function, but certainly energetics and neural function, and all of that is necessary in order to walk a given distance at a given speed.
So to answer your question, what I do is I measure orthostatic blood pressures, which are incredibly helpful. And I think that's why anything that stresses the person in a good way is good. The answer to your question we'll know within a year or so when these two NIA-funded studies, one is at Hopkins, one is at Duke, will conclude, and they specifically are focused on that, is seeing which simple physiological measures and biomarkers at baseline are predictive of how people do from resilience point of view after a defined stressor, such as knee replacement, such as elective anesthesia and surgery afterwards. So we should have that answer. We should have data on that in the near future.
Yeah, thank you. Well, we have about 5 minutes left, and I'd like to open this up for questions from the audience. So yes, speak loudly and we can pass a microphone.
Levels affects health or outcome?
We should talk in the hallway because it's a long answer, but what's coming out of the liver is a measure of stress, and you can decrease those D levels with inflammation as it occurs, which is why we were misled to think that vitamin D levels, 99% of D is bound to that vitamin, excuse me, the vitamin is bound to the protein, and it's a marker of inflammation and bad things, which is why people thought the vitamin D levels were associated with bad. Is there something you can do about the D binding protein levels? Gee, not that I know of, not necessarily that I'd want to do, because it's very much like albumin, a very, very necessary protein for other kinds of functions. So great question. We can talk more about.
Okay.
Let's move over here to, yeah, go ahead.
Just want to give a plug to a paper that just came out in my archive that I'm not an author on, by Gary Churchill and the Calico team. They studied 900 diversity outcross mice that are about 10 times more diverse than we are in this audience. And they did males and females, and they did 6 different diets. And what you'll like, Stephen, is that RDW came out #1 as a predictor of longevity across all of those diets.
You know, you have to look really closely at the mouse posters that are around, but whenever they do a CBC that includes it, yeah, look at RDW in your mice. I don't know about worms, but mice, and you'll find that it acts sort of like humans.
Yeah, and I was heartbroken to find out I have sucky RDW. About
you have
2
a high
months
one?
ago. After
it's
COVID,
anything
though.
over 13.5%.
After COVID.
I'm
See,
sorry, go back to your record.
It was dynamic. It may change. It may get better. Doug.
Hi, Doug Keel from Boston. I was curious if the panel would address the geroscience hallmarks that are not individual options for target, because they're all interrelated, and whether this composite biomarker quest that was summarized in the first paper from the consortium means we must approach this. If you're thinking geroscience, maybe one target is connected to 4 other hallmarks of the aging process. So do we really think that we need a broad composite, or can we find the gold with
Stephen is sighing over here in response to that question. So
RDW.
give him a chance to respond.
Thanks for your question. No. But I'd like to. I mean, there's some persuasive arguments that what's going on in our DNA, the mutations in particular, drive a lot of things that happen downstream. I think that's a persuasive point of view from Jan Vig and Vera.
Yeah,
but I
right.
don't know of one thing that's going to be the font of all aging and target.
Yeah, many of the biomarkers are more consequence of aging, like many things go wrong. Some of them may be more causal, and I don't want to open a can of worms now trying to claim which one. I mean, I have my own favorite one, which would be
This will be the last.
genetic genome stability and epigenetics. But other people may have other thoughts on that. So I think if we sort hallmarks in this way, so then we can say, okay, which one we target. But at the same time, anything that alleviates even one is going to make a difference in human aging. So I think at the first part, maybe we should be very picky. But then for sort of the pure science perspective, yes, it is very, it would be really exciting to find the causal one and just target that.
Sure. We're going to have to wrap up. So quick comment
Very
from
quickly,
George.
I would
and
just
we'll-
say that the choice of biomarkers goes back to the choice of gerotherapeutic. And the choice of gerotherapeutic, I think, what Geroscience 201 will teach us is what we're beginning to learn from Geroscience 101 is that gerotherapeutics that target multiple hallmarks or the target hallmarks upstream that then influence other hallmarks are much more likely to work than gerotherapeutics that are targeting specific pathways way at downstream at the event.
Well, thank you. Well, thanks to our panelists. They did a fabulous job. Yep, good. Very interesting. Yeah. Well, thanks, George.
Thank you, Luis, for this
Sure.
great session. And I'm now inviting the members of the next panel and their chair, Neil Barzilai, who is going to be discussing the NIA
Wake up and smell the flower.
Biomarkers Initiative.
Terrific. Can I have the slides that I prepared? Obviously, we talked about what we're going to say today, and most of what we're going to talk about already was said. So we'll have to have some flexibility, not all, but some of it. So let me ask you, who here actually measured any biomarker on yourself, whether it's epigenetic or the Belsky one or stuff? Raise your hand. Okay, guys, look around, okay? So I think that you're basically healthy people. You seem, probably most of you are younger than your age. And I think some of the reason we measure those biomarkers is so that we can maximize our health. And indeed, I'm standing here now, not later. Later as my research, but now as a scientific director of American Federation for Aging Research that supports this. I'm also an executive in the Longevity Biotech Association that prepares biotechs. I'm also on the board of the Academy of Health Span and Lifespan, but most important, I'm in the council of the Healthy Longevity Medicine Society that Andrea is the president, and that is trying to think, how we take people at any age basically and maximize your health. I should say Eric is not here, but I always have to say exercise, nutrition, sleep, and social connectivity, which is something we can always do. And I guess that our issue. So this is what I want to say as a preliminary thing. This example is preliminary for our talk. This is my Green Age, and when I got it, I contacted Steve Horvath and I said, "You owe me 4 years." Okay. We said why, and I said two answers. First of all, this is my biological age from an EKG. So listen, EKG, if you take it to AI, you get a lot of things. One of the things you get from AI, if you're a man or a woman, by 100%. And by the way, I thought, I didn't have a problem with that. I read the name on the EKG and I know if it's a man or a woman. And they said, well, who's pet? Could be anyone. But AI can tell you, and if you ask a cardiologist, can you look at the EKG and tell me if it's a man and a woman when I hide the name? The answer is no. Also, it gives you the muscle age here, okay? And think about it, it's the muscle age of your heart. But if you get a coronary the next day, it's not going to be the same at the same age. It doesn't depict your risk here, but it depicts the heart. And you saw that I was at the Mayo twice, when I was 58 and when I was 67. And when I was 58, my heart age was 54, which is fine. And since then, what I've done is 2 interventions. I started metformin because I was pre-diabetic. And then before COVID, I started intermittent fasting. So this is pretty much what I'm doing, in addition to exercise and all the other things. So I'm believing for myself that that my biological age actually have changed or have decelerated because of my intervention. And this is what we want to tell ourselves or tell our patients. So that's one argument that and by the way, I should tell you, I've done like 10 tests, and I'm anywhere between 10 years younger to 3 years older than my age. Okay, right? That's, I think we all agree. You can look at all those biomarkers and it's whatever. You can pick what you like, but the average is not always good. The second argument with Steve is that the Grimm age gives you actually a little bit hint in the biology. It's not that they're measuring this peptide, but there's an epigenetic methylation markers that are associated with the action of those peptides. And you see that they're all in the green, except the hemoglobin A1C, which is the red. Okay. So I told Steve, my hemoglobin A1, I understand why it is, because I have 4 SNPs for diabetes. My mother was diabetes. I was pre-diabetic myself. I understand why. Maybe through that, my epigenetic will be there. However, because of metformin intermittent fasting, my hemoglobin A1C was low in the last 10 years, which means this epigenetic clock didn't depict that. That's where I wanted my 4 years back. Okay, so this is just underlying. We can go. I'm not going to show other slides. You can take it off. So what I'm trying to say, you know, there are biomarkers or like EKG that we don't know of. There could be confusing. There are reasons for that. But basically, what we want to focus now is on NIA and its effort to do clinical trials, so we can see not only what are the biomarkers for aging, but which are changing in therapy. So now I'll ask my friends and colleagues here to just briefly say, why do you think you're here, and what do we need more to know? And Steve, start.
Yeah, thanks. I'm Steve Horvath from Wake Forest School of Medicine. Wake Forest was one of 6 sites funded by the NIA's Predictive Biomarkers Consortium, I guess, or network certainly. The other funded sites were the Buck Institute, Mayo, along with Sutter Health that Nathan represents, Indiana University under the direction of Homer Twigg, Johns Hopkins with Dennis Wirtz, at UCLA with Steve Horvath, and Duke University for Virginia Krause. And the purpose of this network was to develop novel biomarkers that predict outcomes of interest in human populations for anticipating trials in the future. So, and importantly, who should be in the trial? So who's at risk based on certain aspects of the biology of aging? Because if you enroll people in a trial who aren't at risk and treat them, they will give you no answer and no results. So that's very important. And also to break away from to complement fluid biomarkers with other kinds of biomarkers. So for example, Dr. Wirtz had cellular imaging biomarkers where he could tell you the age of your skin cells through AI-driven mechanisms to calculate the geography of the cell and its geometry to tell you about aging. So just briefly at Wake Forest are the 2.
Can we wait for that? Because I hope that through what you answer, they also know who you are now. But let me just And yes, we want to focus on, and I want to know about Wake Forest. Brigitte, tell us who you are and what have you done in this consortium?
Okay, thank you so much. Thanks, Steve, for already giving a great introduction. So I work here at the Buck Institute. My focus is, at cellular senescence, and then using really technologies to really dive deep into cellular senescence, secretomes, and then together with Judy Campisi and her expertise over the many years, so really having a discovery platform, using a technology to discover, for example, the SASP, and then finding it in tissues and more importantly, biofluids. So I think our group has really focused on the combination of technology, discovery, and then really finding that verification. I loved earlier some of the slides shown, right? You go from, discovery to verification, validation, translation. That's my focus. Mm-hmm.
Nathan.
Well, thanks, Nir. It's great to be here. And thanks so much for organizing this symposium. I think it's outstanding. I'm not sure how much more we have to say, right? A lot of the questions and issues have been raised, but I had the pleasure of participating in the Predictive Biomarkers Network and focusing on senescence as well as a mechanism of aging and kind of the poor man's approach to Dr. Schilling's, where we really exploited the secretome of human cell types that we think contribute to circulating protein concentrations. We talk a lot about biomarkers. We don't talk a lot about where those biomarkers are coming from. And we think senescent cells are a plausible source, either directly or indirectly, and we can get more into that. And through this network, we've been able to look cross-sectionally at the association between these biomarkers in key kind of credibility criteria, I think, such as associations with chronological age, looking at the relationships with clinical manifestations of advanced biological age, whether it be frailty, disease, rehospitalization events in different patients. And then you kind of move from that cross-sectional and short-term longitudinal approach to looking at the predictive ability of these biomarkers for longer-term outcomes. I think that's the next step. And the big step is longitudinal changes in these biomarkers that may change with different clinical manifestations of aging. And ultimately, of course, looking at interventions and how they influence them is really the next step.
So terrific. So Steve, so back to you. You started to say Wake Forest. So what have you discovered?
Well, the two areas of focus of Wake Forest, one was in the transcriptome of the monocyte. This is work led by Jingzhong Ding in the MESA cohort. So these materials are available and there are the transcriptome of monocytes is available in about 1,200 participants of that study, and they're redoing that currently. And found that transcriptome profile associated with chronologic age was also associated with both the prevalence and incidence of multimorbidities, which I think is a very important observation because it sort of substantiates part of the geroscience hypothesis that single mechanisms can drive multiple age-related outcomes. So a very important set of observations. The other aspect was a slightly different angle by Anthony Molina, who's now at UC San Diego, looking at what in the blood affects mitochondrial function in standardized cells. So looking at blood biomarkers that might be tied exactly to mitochondrial function. And there are some promising results there that are now being tested in the SOMA study.
Nathan, you did the CLARI study as part of this initiative, or that was the CLARI study?
Yeah, that was a bit of an administrative supplement to another grant, but yeah, so we've been using kind of a panel of about 40 biomarkers and testing them in different contexts and being able to show that they're highly predictive of outcomes such as loss of mobility in older adults and functional limitations, some measures of pulmonary function in different disease contexts. And then most recently, we've looked at two different cohorts, one which is rather unique. So Mayo Clinic has a biobank of about 56,000 participants, individuals who have agreed to contribute blood and blood cells to a research database that's linked to their electronic medical records. 24,000 of those individuals are over the age of 65. So we actually took 2,000 of those individuals and measured these biomarkers of senescence, and we're able to predict mortality in that cohort. And what's unique about that cohort is that they only had 0 or 1 medical condition at baseline. We had about 7 years of follow-up. So our first efforts to see if those biomarkers also change in response to intervention was taking advantage of the calorie cohort, another outstanding resource that's available. So these are individuals that were assigned to either an ad libitum diet or caloric restriction for 2 years. The goal was 25% calorie restriction. They actually achieved about 12%. And as much as you say, well, maybe that didn't do anything, their cardiovascular risk profile and their metabolic profiles looked remarkably improved after the end of that intervention period. And in that cohort, we did show that these markers of senescence did decrease in the blood. And then we had just a very small sample of participants who provided adipose tissue. And we showed that a gene set for senescence was decreased in their adipose tissue samples as well.
And Brigitte, what did you find? So you're not the clinical investigator here. What was your project and what did you find?
Thank you. Yeah, we connected with several people who had access to clinical samples and also specifically with Luigi. And we looked basically in the BLSA and also in Chianti and saw that a lot of the aging markers were actually indeed senescence markers, which was basically the proof of principle for us that to really go after senescence markers. And I really think that that has really propelled us to do that. So since then we have deepened our senescence signatures from different cell types, even Schwann cells, but also more recently from, inducing senescence, say in explant tissues such as ovary and muscle, and then also in organoids, say brain organoids, really building these senescent signatures that we are now really going and validating in the plasma workflows. I wanted to also say again a little bit the validity of having both mouse models and human work together. So in my lab, we connect a lot with collaborators where then, for example, we do some plasma work. There's kind of a human aging tissue collection and even a mouse tissue collection, and kind of looping all of these things together has given us a lot of candidates that show up across many tissues. So yeah, I think we're trying to build these signatures, and in fact, a lot of our signatures do indeed overlap.
We steal from you.
So was this? I want to move from this consortium, but was this consortium successful, or what are the lessons that we have? Okay, I'm not hearing. There's incremental things I'm not hearing, but what did we learn, Steve?
Well, I think I do remember the first time everyone got together, how much excitement there was for people who had traditionally done a lot of work in mice, meeting folks with humans and arranging collaboration. So I think a meeting like this has a great power in convening people of similar interests, but perhaps in different kinds of networks and resources. Another thing that we learned was how challenging it was to find interesting sets of human samples that could be evaluated by everyone in the consortium at the same time. So the fluid biomarkers and the epigenetic clocks worked well together and could be done, but things like cells saved for transcriptomics or tissue biopsies for imaging, not so easily. And so there is a need going forward to be somewhat systematic in banking tissues that can be used for a wide amount of omics. And the other lesson was the need for intervention studies. Calorie was one, caloric restriction, a really good one, but that was about it. It was very hard to find studies with human samples banked of interventions of great geroscience interest that could be used to compare the performance of markers by different groups. So.
Yeah. Right. Great.
Or maybe I start. Well, I think one of the bigger successes of the predictive biomarker studies, I thought, was really establishing these collaborations, because, for example, a lot of us now collaborate together and do studies together. So I think, and I mean, maybe this was a small version of what this meeting could be, right? Really, I feel really collaborating. And for example, for us now, Steve's group and us, we have a new grant together, right, where we get muscle tissues from Wake Forest, and with Dennis Wirtz we work together. And so I think really connecting aging biomarker interested people, that's a first success, I thought. And yeah, I think, yeah, that was something very important to us, I feel.
Nathan, say what you think, but also I want to know, as a follow-up, is the technology different now? Like, if you had to make some of those studies, would it be different?
Yeah, that's a great question. I think the informatics side is so remarkable. And just thinking about my own lab, when we do more kind of cell-based research or animal-based research or human research, our ability to go into publicly available data sets for confirmatory or validation types of analyses. So I do think the technology has rapidly advanced. And of course, we're all now using kind of omics approaches, digital spatial profiling, other things, just to kind of really better understand the biology we're interested in. And I guess that would be my answer to your question here about what have I learned. And I think, you know, we're in this swell of energy and momentum in geroscience, but geroscience is still a hypothesis. And because of that, I think we need to pump the brakes a little bit and also make sure that we're keeping a focus on the basic science and understanding that science. Yes, we want to go into humans. Yes, we want to move interventions forward, but I think we still need to really better understand that biology. And part of this biomarker work should really be developing better biomarkers for the different hallmarks and the emerging hallmarks that we're so interested in.
So Steve, there's one great NIH success, and that's the grant that was submitted to the NIH for the biomarker of TAME, which basically was written by Jamie. Where's Jamie? Jamie Justice, and got a perfect score, if I remember right. Just 11? 11. So maybe tell a little bit about the effort and how is that a guiding thing for us?
So for those of you who don't know, and I can't believe with Nir's efforts, anyone here doesn't know that TAME is a sort of a concept for a clinical trial to test whether metformin slows aging, as represented in the delayed onset of several age-related comorbidities. And the point of the study was to test this proposition, for which there's a lot of epidemiologic evidence that it might be true, and certainly equipoise on that. And metformin was of interest because of its safety and how inexpensive it would be to deliver. But a bigger, not a bigger prize, but another prize of doing that study is to provide a touchstone for biomarker development and biomarker validation, because you heard Steve Cummings talk about surrogate outcomes. And in order to validate a surrogate outcome, you have to have an intervention that affects the outcome that you want the surrogate for. And until we have that in geroscience, it's impossible to validate surrogate outcomes. So the grant, when we thought we were going to be getting funding for TAME, we put in a grant to NIA to fund a biomarker validation discovery, which has some candidate biomarkers that we selected from the literature, but mostly set up a repository and platform because we saw how quickly the biomarker world was moving, and what we put in would not be what we'd want to do 8 years later when we were ready to do it. So that was very persuasive to the reviewers. They absolutely got the need for biomarker repositories in an intervention setting of an intervention that has geroscience promise.
And interestingly, there was no mention of the technology. We said when we'll get there, that when we're writing the grant and until we get to the technology was so far ahead that we'll have a committee that will decide that. And here's the committee here is in this room. Okay, so let's talk about the future in your labs, and also so let's start with that, and then we'll do a round of, you know, if you had a wish, what it would do.
Maybe I could say something.
Sure.
So we were fortunate here at the Buck Institute, and that is actually together with Steve's group, to be now part of the Senescence Network, SenNet, which is out of the Office of Director, but is actually supported by NIA also and executed by NIA. And actually George is part of that also. That has given us an amazing group of people, and really we just had a big meeting in Minneapolis, and where everybody came together. And so now this has started to result in tissue interchange, right? And this is all human. And so I think that is a bit maybe the next step of what was the predictive biomarker. And of course, the same ideas are there. What are biomarkers? They are looking a bit for senescence-derived biomarkers, but I think potentially what I would wish, as you say, I think one of the aspects and we already have this in phase 1 is interventions by senolytics, and that initially may be an explant tissues, but that's human explant tissues of different ages where we induce senescence. We have actually done that now and found a lot of our omics biomarkers, which was super exciting. And now we're using senolytics in that. And I think, you know, and then one could look at all of this in the different tissues with the different teams, all the power that those teams have, and they have, again, many omics in this group of people. So I think I'm very excited about that.
Nathan.
Yeah, I would share the same comments that really leveraging the newer technologies to better understand the biology that we're trying to study, and using that biology to better inform the biomarkers that we're measuring. And I guess, you know, part of this too is just recognizing that I'm not sure how many times the word senolytic has been said today, but it's almost as though we have this go-to medicine cabinet with these drugs that have been shown to have senotherapeutic properties. And I just think, again, we're so early in this game that we need this discovery science to better inform biomarkers and to better inform drug design and drug discovery. And when we go into humans with those emerging drugs, having biomarkers to tell us, are we engaging this target or not? And I'm not just picking on my own field of xenotherapeutics, but whether it's mitochondrial enhancers or things that stimulate autophagy, we need to understand this biology in a way that can inform us, is this the right treatment for the right person at the right time? Are we engaging that target in a meaningful way? Is this the optimal dose? Is this the optimal dosing regimen? I just think, and I'm not saying that to be negative at all, but I just think let's not rush things too much.
Right. Actually, if I may comment briefly on that, I actually 100% agree with you. And when I just said senolytics, my mind is actually highly open to anything, right? Any drug. And I really think that molecular measurements can really inform what might be a good drug, and maybe working more together also with medicinal chemists and computational drug design. I mean, that's like my vision, really using all the things we learn.
So we move now from biomarkers to curing aging. I see where.
I mean, but we need the biomarker research, right, initially. Mm-hmm.
So I think I agree with all of that. Another thing is there's a big gap in our understanding of hallmarks of molecular aging in a cell and the phenotypic changes that we would hope to see in a clinical trial. And so it's very hard to design a study rationally without filling in the blanks in between, because in the best case, I think right now is for senolytics, because the mouse work that's been done with the mice that you can affect the senolytic burden gives you some indication of what you'd hope to see if this were successful in humans. We need to know that because along with knowing which individual has what problem in their aging biology, if you could say that, or what's the geroscience opportunity for an individual, and are they at risk for the outcomes that are associated with that that particular feature? And because those are the people you'd want in your trial. Again, a lot of the early criticism, a lot of the vitamin D trials is they were all involved people who had fine vitamin D levels. So why would you expect them to have any benefit? But we've seen as those have accumulated that the point that Steve Cummings made about them holds. The other aspect is there's a reverse part of this that I think would be fun. It may be premature. So I'm part of, along with George and Tamara Ciconia and Jim Kirkland, lead something called the Translational Geroscience Network, where people doing phase 1 and phase 2 trials of various things they think may be important have agreed to share data and compare notes on how that reacts. I think the inverse of that would also be valuable, which would be something like a gerotherapeutic, a geroscience testings program akin to what is in the NIA mouse models, where people who are networked to look at repurposing and behavioral interventions to collect materials in a standardized way that could be compared across biomarker platforms to see what is responding to what and in whom. A lot of this is just an empirical problem. We just need numbers and experience to be able to sort this out. I think we can think about it a lot, but until we start collecting data, it's going to be hard to know if we're heading in the right direction.
So I totally agree with this comment that we need more from the hallmark, and we need to personalize everything that we do, because we need to choose the right drug to the right person, right? If we have 1,000 drugs out there, how are we going to use them? And wouldn't they be we assume, Brian Johnson assumed that he's taking 115 supplements and drugs, and they're additive or synergistic to each other, but actually, his Green age at the same meeting was just 4 years. It's like was mine. So he must be antagonistic also, right? So there's going to be antagonistic treatment. But I'll tell you my marketing now from the Longevity Biotech Association. All those drugs that we're talking about potentially repurposing, whether it's metformin, GLP-1, SGLT2 inhibitors, etidronate, they all actually change all the hallmarks of aging. That's why the hallmarks of aging are always, we have lines between them. And the concept is that no matter what you do, no matter what the drug is doing, it takes an old cell or an old organ or an old body and make it younger, so it fixes lots of things. We used to fight, what's the mechanism of action of something? Well, yeah, they're doing everything because they're doing something to make it. And I think this concept is important, because once you're doing that an effective other, it means that the drug, the target one hallmark, can be relevant to disease that is more relevant to another hallmark. Okay, and so I think we have a strength from maintaining this hallmark and their halos. But for us, I think we have to dive in and find, 201. Okay, anything else for the future before we're opening up for questions? Okay, so please use the talents here and ask your question, please.
You're interested, you mentioned, in patient selection biomarkers. And those patient selection biomarkers are for individuals that would be going through clinical trials for prospective therapeutics?
Yeah, it's no different than measuring blood pressure for treating an antihypertensive drug or LDL cholesterol if you have a statin drug. People are selected into those trials. It's especially important right now. because if there's ever going to be an FDA approval of something in this, it has to affect the way someone feels, functions, or survives. Okay, that's their criteria. And so we need to know if we're going to get to that, we're going to have a very large trial or a pretty large trial. And people have to be at risk for whatever outcome of feeling, functioning, or surviving outcome. Because if they're not, that's just wasted money in putting them into the study.
So you're thinking mostly about enrichment in those trials, as opposed to the possibility that some of those biomarkers might eventually make it into companion diagnostics.
Say that last bit.
Companion diagnostics.
Oh, oh, that's interesting. I think eventually, perhaps. I think not initially, but perhaps eventually. Yes.
Okay, I have a question. So there are two words here that are kind of talked together, but not truly connected, which is on one side, we talk about biomarker of aging. On the other side, we talk about the hallmarks of aging and the mechanism of aging. And I think that at this point, the connections are very thin. There was some initial attempt to look at them from the Steve Horvath group. And but then, do you think that this is worthwhile to do? Do we need to measure, autophagy in human? We need to measure, to have a measure of DNA repair in human and connect them with the biomarker. So the biology of aging is really reflected by the biomarker.
Well, thank you, Luigi, for the question. I think for our own group, we have found a lot of things that I think we think a little bit differently around now. For example, in my case, looking at senescence-derived biomarkers, we have found the importance of course, the secretome, but also the extracellular matrix, which indeed can actually determine fate of other cells. And this may have to do with bystander senescence or also traveling of markers through the circulation to other organs. But even in a study with Luigi, we are finding that there's a lot of splicing in cellular senescence, which I think that's important biology to find out about, because maybe if that could be something interesting or to be treated, that may be potentially an even different mechanism. So I think biomarker research, whether it is discovery or application, always informs biology of aging, I think, because even when you say you treat with a drug now and you monitor certain biomarkers, imagine a panel of biomarkers, maybe it only has response to some of those biomarkers. So then you already learn again about the biology. I mean, as you said, I think these things are super closely related to each other.
Well, they can be related. I totally agree with you. And that's because we're both doing proteomics, right? So look, I'll show later, but the biggest thing that I see in proteomic is breakdown of lots of things, breakdown of collagen and breakdown of white cells and breakdown of, and so you think of it, okay, there may be several ways to stop the breakdown. I don't need, so I don't need to understand what the hallmarks are, as long as everything that I do stops the breakdown. I think that's kind of the dissociation. We don't necessarily, we can make a lot of progress now by targeting the biomarkers without understanding the biology. But I'll give you another example, and then I'll show later, because we've been collaborating too, and we got similar results. But remember that the SASP that you had, okay, half of them increased in our patients, but half of them have decreased. So my interpretation here is that the SASP, the decrease bothers me, because you can have SASP that come up and stay in the tissue, but they shouldn't decrease the level. So whether our proteomic is measuring the SASP totally, I don't know, but am I saying we don't have to understand the biology? No, not at all.
No. Well, SASP is not all bad. That's also important, right?
You said that. I'm not telling Judy that.
No. But anyway, I think, Nathan, I'm sure you want to comment also on, I mean, I think learning about I mean, in any study, I think you learn about biology, and everything we do, we add to our knowledge base, I feel. And trying different drugs of different types, I'm interested in also because they may find different mechanisms. We talked earlier about combinations also.
Yes. It's all clear, you know how to do clinical studies? Eileen.
So thank you very much for this whole discussion and also listening. So I'm Eileen Slagboom from the Netherlands. You know, it's not the only European, but we are outnumbered. What I like very much also in Steve's remark and Vera's remark is that when you measure this double-strand breaks, then you're measuring the stochasticity of aging. And that's also the way I look at it with many of these hallmarks, is that when we are taking all these systemic factors to investigate our risk or our vulnerability, it might be smart to add some of these stochastic markers in aging, because individuals may have many more breaks, and that might be a sign that also other stochasticity is going on. And that will really, is very hard to measure, but it will also disturb your original systemic markers very much.
How do you do it in a clinical study? What are the markers that you could do? I mean, it's a good biological question, right? It's less of a clinical.
Well, it's less of a clinical, so I'm also not doing it. I'm having this
brainwave
brainwave here, actually.
Yeah,
that
it's
I
a great
realized,
idea.
Yeah, so when we talk about hallmarks of aging, we always think about the causality and etc.
Right.
But if you think about biomarkers, maybe we have not enough stochastic markers to accompany, but I'm convinced that you need different types of markers anyway, also in the clinic. Otherwise, you cannot type a human. But I think that the part that we never type is the most stochastic part. That's what I want to say.
Absolutely. Hey.
Yeah, I actually wanted to follow up very much on what Eileen was just saying, because there's a very related thing that's been on my mind recently. I think that we have to talk about the possibility that there is an entire universe within the epigenetic space that we're not looking at right now, namely how the cell manages to make epigenetic age change as slowly as it does. I think, you know, in the same way that we look today at loss of proteostasis, you know, which is obviously a big deal, we maybe should also be looking at loss of epigenostasis. You know, how the body does this. So I guess many of you will have seen the paper that came out of the Convoy lab recently, which looks, I think for the first time, at the variability of epigenetic age within an individual at a particular time. Just a first step in this general direction. But the fact that there is such an increase with age in that variability tells me that the stochastic aspect that Ellen was just mentioning applies really heavily within epigenetic state, and nobody's looking at this. So maybe that's a whole new dimension to how we can develop better epigenetic clocks.
Can I, if anybody wants to comment, but can I ask the poster presenters to come here? We're finishing in a couple of minutes, so you can come and be ready. Anybody wants to say anything about Aubrey's, proteostasis, epigenesis. Okay, good. Thank you, both of you. That good talks. I hope that that you kind of learn where we are. What are the challenges? What do we have to do? But overall, I think there's a great optimism. I certainly think that we went from half a cup poison to cup that's almost full. So thank you, everybody, and thank you, the organizers. And also, please remind me to tell you at the end of that everybody needs to go out and have a picture taken from all of us. So
yeah, so now it's flash talks. It's typically the most exciting part of the program. And I'd like to introduce Joe Betzlacroix, who is CEO of Retro, who will chair that exciting session.
Hey there. Let's see. Hear this. You turned it off.
Hey there. Okay, I'm Joe, and we have quite a lineup here along the wall of people who are going to give you 60-second talks. I'm going to be very strict about this. I will just start slowly pushing you off the stage if you exceed 60 seconds, which is going to be timed by that clock right there. Okay, without further ado, we have coming up Andrei Tsetiskov talking about G quadruplex DNA and RNA and cellular senescence. Go.
So I'm Andrei Tsvetkov, neurology, UT Houston. And in my lab, we are working on understanding how G quadruplex DNA and RNA regulate cell senescence. G quadruplex is a 4-stranded DNA or RNA structure that falls from single-stranded guanine-rich DNA or RNA sequences. These guanines self-associate and form a quadruplex. Here's how a G4 looks like compared to the duplex DNA. So normally G4s are located near the replication sites or gene promoters where they modulate replication and transcription. And these proteins bind and regulate G4s. So under pathogenic conditions in which G4s are overly stabilized, they can inhibit transcription and translation and other processes as well.
And so these are all posters, and you can go out and see them
Yeah.
on the poster boards, because obviously there's more. Okay, we have Raghav Sengal talking about multidimensional aging biomarkers.
Go. Okay. My name is Raghav Sehgal. I'm a PhD student at Yale studying multidimensional biomarkers. And I just kind of want to state the obvious that aging is multidimensional. All of us age very differently. Some of us will get cardiovascular disease, others will get neurodegenerative diseases. And if that is the case, why the hell are our biomarkers unidimensional? Why are we all lumping them together, as Steve says it? We are all trying to predict chronological age, time to death, pace of aging using, well, epigenetics, other sources. And I believe that we should be sort of splitting them up. And the reason, and for that very purpose, I have been building multidimensional biomarkers of aging, such as systems age, which can quantify organ ages from 11 different physiological systems using a single DNA methylation blood draw. And the same is true for chronic stress age. Come to my poster, number 2. And I left with 3 seconds. Thank you.
Wow, congratulations. Okay, we've got Austin Argentietti talking about proteomic aging.
Hi, everyone. My name is Austin Argentieri. I'm at Harvard Medical School in Oxford. So if aging is truly the largest determinant of most common chronic diseases, then we should be able to quantify a biological aging signature that's associated in humans with most major chronic diseases. And that's exactly what we've done. So in a study population of over 47,000 individuals with proteomics data and decades of linked electronic health data, we developed a proteomic age clock using machine learning, and here's what we found. Accelerated proteomic age is associated with nearly all major chronic diseases and all-cause mortality, and also not shown here, several cancers and multimorbidity. And accelerated proteomic age is also associated with nearly 25 biomarkers of aging and other physical and cognitive and functional decline measures. So we conclude that proteomic aging is a possibly powerful tool for preventative medicine and for identifying different preventative and monitoring strategies for multiple chronic diseases simultaneously.
Perfect. Okay, we have Zane Koch, somatic mutation as an explanation of epigenetic aging.
No animations, huh? So if we're here to study aging and understand how to promote healthy human lifespan and develop interventions, we want to understand the way we are understanding aging. And a lot of people have talked about epigenetic clocks and the epigenome, and this has kind of led to a succession of epigenetic-based theories of aging. But in my opinion, we still don't know why the epigenome changes with age, and even more so why those changes predict mortality. And so it was my goal with this project to answer some of those questions. And we do this by analyzing somatic mutation and DNA methylation profiles from around 10,000 individuals and find that somatic mutations associate with pervasive DNA methylation remodeling. We build mutation clocks, which predict chronological age and explain almost half the variance in epigenetic age. And come to poster number 4 to discuss the implications of this relationship between somatic mutations, methylation, and aging with me.
Woo. Here we have Xiaojing Yang.
So Xiaojing's over there in the green sweater.
Oh,
so
okay.
If anybody has questions, ask her, not me. My name's Abigail. I am here together with Xiaojing to present on behalf of Epimorphy. Anytime any of you publishes something on interventions that could influence longevity, the Brian Johnsons of this world are immediately taking it on. Now, the challenge is, how do we collect all of this real world information and real world evidence to refine our own, number one, ability to understand which biomarkers are accurate, are relevant, and what is exactly the impact on everything. So what we have is a direct-to-consumer platform, and we believe that using our direct-to-consumer platform could potentially be a way to enable us to make a significant leap forward in aging research by providing that real-world feedback very quickly so that we are able to sort of say yes to this, no to that. Come and find us at poster 5 and we'll be able to talk more about this.
Great. Okay, we have Lucas talking about histone mark age. Cool.
It's still my slides. Sorry, you can come to my poster and show you interesting.
Just skip. Yeah, keep going. Oh my God. Oh my God. Oh my God.
Okay, that's me. So I think we can all agree that DNA methylation is highly connected with aging. And this is not true, this is not only true in humans, but as was shown recently, also across mammalian species. However, there are some non-mammalian species that do age, but do not have any meaningful DNA methylation. So for instance, C. elegans and Drosophila, throughout evolution, they lost DNA methyltransferases along the way. So we searched for an alternative to DNA methylation, e.g. histone marks, which are virtually ubiquitous. We were able to build highly accurate aging clocks based on cheap-seq histone mark data for 7 different histone marks. I consider the definition is not fantastic, but more surprisingly, we were able to build a pan-histone mark that shows how we can measure epigenetic aging with a single formula. Lastly, it's available on PyAging if you Google that online, and this is a link to the preprint. Thank you.
Perfect. Tsuyoshi will talk about phenome-wide Mendelian randomization study. What? Okay, I'm just going off this list here. Who's who?
I have no slide. I'm Tsuyoshi, come from Japan, and our aim is to identify potential causal factor associated with longevity using a statistical approach called Mendelian randomization. Our results showed that longer longevity was associated with higher education, higher HDL cholesterol, and increased lung function, and found that shorter lifespan is associated with obesity, hypertension, poor self-rated health, smoking, high LDL cholesterol, and watching TV. Thank you.
Okay. We have Daniel Simpson talking about PRC2 age index.
Thank you. All right, so what's been talked about a lot here is the biological relevance of epigenetic clocks and the CPGs utilized. So what we've developed at Vittorio Sebastiano's lab is the PRC2 age index. So looking first at PRC2 bound regions, which has been known, and we've seen that actually, oops, I was expecting animation there, never mind. They low methylated regions associated with PRC2 gain methylation with age. And if you actually sort these regions based on PRC2 binding, you can stratify samples based on age, passage number, and even look at interventions such as calorie restriction and OSKM-based rejuvenation. And we actually see a significant overlap of these top low methylated regions between different tissues and an enrichment for development, which has interesting age-based theory context. And I'd like to thank Mahdi Moqri and Andrea Cipriano as my co-host.
Awesome. All right, we have Denis Kotasevich talking about machine learning clock things.
Hi, I'm Denis Kotasevich at UC Berkeley, and in this project, we examined the application of ensemble machine learning towards epigenetic clock development. So most epigenetic clocks are often trained with a last net regression of individual CPG sites, but some recent evidence has suggested noise reduction benefits of training on principal components derived from this instead. The really substantial data reduction provided by principal component analysis may also enable the implementation of more advanced machine learning here. And so we developed a pipeline capable of training 3 clocks. So first, the traditional approach based on last net regression of CPGs. Second, principal component analysis followed by a last net regression of the PCs. And thirdly, a superlearner ensemble predictor based on the PCs. And we compiled a novel childhood clock training data set, and as well as several testing data sets. And we found with the superlearner-based clock, we found improved fit in chronological age and complex samples, more precise defect estimates and longitudinal analyses, as well as higher agreement between cross-array replicates. Oh, and I'll be at poster number 9.
Nice. Super on time. Thank you. Varun Dwaraka, omic age.
Thanks, Joe. Can you hear me? Okay, perfect. Well, thank you, everyone. We're delighted to be here representing True Diagnostic Incorporated, and along with that, Jessica Lasky-Sue's group here. I am Varun Dwarka, and what we'll be discussing at our poster today is Omic MH, an integrative multi-omics approach to quantify biological age with electronic medical records. This is actually just to test your speed reading, but really focusing on this is those bolded aspects, because what we were able to do is taking a lot of the framework developed by Dr. Steve Horvath and your group, we were able to actually integrate multi-omic information into a single composite score. But what is really important is how well it is able to function as associating to different types of diseases. So if you come to the poster, we'll go through this in detail, show the biological reasons for this, and this is how the poster looks like. And there is a preprint available on medRxiv or bioRxiv. So please check it out.
Great. All right, we have Tarif Awad talking about using proteomics to screen for age-related illnesses.
All right. Thank you so much. My name is Tarif Awad. I'm with Olink Proteomics, and I'm going to quickly describe work that was done by a group at the Science for Life Laboratory in Sweden, supported by Olink. Here's the very briefly the work described. Close to 2,000 patients had their plasma proteome profiled using the Olink discovery platform, which, by the way, at the time they used the earlier version of the discovery platform, it was only close to 1,500 proteins. Our current platform offers 5,400 extremely well validated proteins. So it's an awesome way to discover biomarkers. The focus of the study really was to look at across a variety of different cancers, and we looked at 15 different cancer types across multiple ages. They're shown right there. And what we found was signatures that are specific to cancer types. Come to the poster and I'll tell you more. Thank you so much.
Perfect timing. Okay, we have Max. We have Joran talking about lipid aging clocks, I think. No? Who? 12 is not here. Okay. Switching to 13, we have Saher.
Yes.
And talking about epigenetic age acceleration at birth.
Okay, so hi everyone. We compared prenatal drivers of epigenetic aging among newborns in 2 cohort studies, Project Viva and Chamacos. Epigenetic clocks have largely been applied to adults. More studies on early life epigenetic aging are needed, especially in diverse populations. Our 2 cohorts have very different sociodemographic characteristics. DNA methylation was measured in cord blood. We estimated epigenetic age using 3 different clocks, and then tested associations between maternal newborn characteristics and epigenetic age acceleration at birth. In both cohorts, epigenetic age from the Bolin clock was highly correlated with chronological gestational age. Preterm birth and female infant sex were associated with decreased epigenetic age acceleration. Associations with other characteristics like maternal smoking, parity, and birth weight varied across our cohorts. If you have any questions or if you would like to learn more, please stop by our poster, which is number 13. Thank you.
Nice. Okay, John Ralston on something unpronounceable. No, I think that's right. Yeah.
Okay, so at Geroscience, we are developing something very pronounceable, non-invasive bioelectronic devices to treat neuromotor conditions. And our initial commercial focus is on age-related balance decline, which actually starts as early as 40, triggered by age-related changes in your vestibular balance organs, which in turn disrupt your entire balance system, are also linked to cognitive decline. Geroscience has pioneered the development of Fibrata wearable sensor and biomarker technologies to quantify all the key components of those balance impairments and to develop a non-invasive bioelectronic device and electrical vestibular stimulation treatment protocol that's now been shown to deliver very large and persistent neuroplastic restoration of balance in populations from 50 to 90 years of age and to potentially slow the related cognitive decline. More details, and I'll give you back 5 seconds at our poster number 14. Thank you.
Thanks. Okay, Fibrata wasn't actually that hard to pronounce. Okay, Oscar Coltel will talk about lifestyle factors.
Is this? Yeah. Hello, I am computer science professor at the Jaume I University at the East Mediterranean coast. We analyzed the association between lifestyle and grim age in a Mediterranean population. Spain is the second country in highest life expectancy after Japan. We address 3 different aims in the 3 populations. As a result, we found a strong correlations between grim age 1 and grim age 2, association between lifestyle variables such as smoking, diet, education, and so on, with grim age acceleration, and finally we found
Great. You can read all about that on that poster out there, which will be very accessible to you. Okay, we have Dolores Correia talking about Mediterranean diet.
Okay, thank you. I'm Dolores Correia. I'm professor of nutrition and genomics at the University of Valencia in Spain. In nutrition, the DNA, the telomere length is an indicator of aging, and there are a lot of studies analyzing this indicator, but there are some bias because there are very different methods to measure this, and we thought that methylation, the DNA methylation biomarker for telomere length will be an interesting biomarker. Then we tested the effect of the real Mediterranean diet in this biomarker, and we obtained valid results. First, we validated the biomarker. Second, we analyzed genetic SNPs in candidate genes and also at the US level, and we obtained a protective effect of the Mediterranean diet. Thank you very much, and come to see my poster.
And eat lunch soon. Thank you. Okay, we have Keijun Ying talking about causal aging biomarkers.
So, hello, I'm Albert Keijun Ying. I'm a PhD candidate from Harvard Medical School. So one of the major limitation of epigenetic clock are lack of causal information. So here we use the epigenome-wide methylionization to identify the potential causal CG site that have potential causal effect to lifespan and health span. And we've ended up finding several thousand of them. And then we're using this information to train a novel kind of clock. We call it consulting-rich clock. And we were able to separate the potential damage and adaptation. And we were able to actually, we can build a clock we call adapt age. It represents age-related adaptation. It's actually, if you have higher adapt age, you have a lower mortality risk. And then what's the next step? Next step, we want to use this information to find novel intervention. So next, we compile the current largest database for the biological age. We call clock base. We have 30 clock, 7,000 data sets, 300,000 samples, and we use this.
So much, so many, incredible. Yeah. Okay. So we have Yoshihiro. Is that right? Is that you?
Takuro.
Oh, Tako. Okay. Oh, sorry.
Yeah,
okay,
no problem.
Takuro Kobayashi talking about alcohol.
Okay. Okay. Hi, hello everyone. Our study focuses on the causal relationship between alcohol intake frequency and their particularly total testosterone levels. In this study, we use the Mendelian randomization MR method to analyze causality based on UK biobank data. This method relies on the principle that genes combination occur randomly at conception. Our study indicates that lower alcohol intake frequency caused genetically lower testosterone levels. In addition, we analyzed opposite MR. Interestingly, testosterone didn't associate with alcohol intake frequency. Moderate frequency of alcohol intake may be necessary to maintain testosterone. If you have any clinical observational study with unclear causal relationship, we may be able to help. Thank you.
Okay, thank you. Perfect. Okay, here's Yoshihiro, actually.
Hi, I'm Yoshihiro Kihata from Juntendo University. Our team is doing aging research in Japan. Oh, sorry. Testosterone is an essential sex hormone. Testosterone are influenced by many factors, but it is difficult to assess the direct effect of body composition due to other confounding factors like fitness or supplement or food. So we did first Mendelian randomization method have been applied to investigate the potential causal association between body composition and testosterone levels. So this is the result. Fat mass was negatively associated with testosterone. On the other hand, fat-free mass, it means muscle mass was not associated. So this MR method suggested that muscle. That's great. It's good.
Okay, we have two left. Only two, unfortunately. Alexandra Binder talking about epigenetic clocks.
Yes. All right, thank you. So there already has been discussion today about the value of reliability when we want to accurately inform risk management using epigenetic clocks. So for example, if we're looking at the same individual, if we look at them across technologies, across assay batches, or even short periods of time like a day, we would hope to get a very similar estimate of their epigenetic age. So in our poster, we present a newly developed tuning method, which imputes site-specific DNA methylation across the epigenome based on methylation patterns that are more reliable across technical artifacts and batches. So we demonstrate that using this tuning method, that this markedly increases the reliability of the epigenetic clocks, maintains the ranking of estimates across individuals, and also increases the strength of associations with mortality. So given this method estimates site-specific DNA methylation, it can be synergistically applied with other methods, such as those mentioned earlier, the PC method, and other epigenetic clocks as developed. So thank you.
Perfect. And last but not least, Ake Lu talking about universal DNA methylation.
Hi, my name is Akeru. I work in Steve Horvath's lab at Altos. I'm going. Okay. So here I'm going to introduce you the details of the third generation epigenetic clocks, also known as universal pan-mammalian clocks. Our clock can predict age from various tissues, from human mass, dogs and even bowhead whales with correlation over 0.96. And as Steve mentioned, that clock has potential application in predicting human mortality risks. And our other novel finding concern age-correlated CPGs. Here we find that age-correlated CPG, like gain methylation with age, are enriched in PRC2 binding region, but not PRC1. So come to my poster, I will show you more details. Thank you.
And here,
hello, I'm Robert Cargill with Quadroscope Venture Fund. I'm a venture partner. Oh, I guess I am in here. I don't know if my slide got in time or not, but yes, indeed. So I guess you might consider us slicers. We are very interested in being able to detect changes in an individual person's epigenome over shorter periods of time. So we would love to see more data like what Michael Snyder has done, many measurements within a period as short as a year. And towards getting there, we believe that one of the most important parts is to have the availability of very inexpensive epigenetic sequencing. And in order to do that, we're currently evaluating a technology for a solid state nanopore. So if you have, if you're aware of data like Michael Snyder's, many epigenetic samples within a year, or if you have an interest in solid state nanopores, please come see me.
Great. Are there an infinite number of unofficial additional poster talks or what? This is last, right? Okay.
I'll be very quick. I'm Jose Navarro Betancourt from Quadroscope Venture Fund, and come to poster number 22 if you want to talk about the approaches we are exploring to build a continuous health monitor for molecular biomarkers of aging. These approaches include measuring mitochondrial membrane potential and nanopore sequencing using a microfluidics approach. Thank you.
Cool. Thank you. Nice and short. So just before we all head off into Foodland, I think Allison has a very quick announcement about a group photo.
I know that we're all really, really dying for lunch. Thank you so, so much for these wonderful presentations. But before that, please head outside in front of the main entrance. We'll be taking a group photo. We have many herders here that will very politely but gently move you out of there, and then we'll break for lunch, okay? Not supposed to be announced. Oh.
Okay, we are nearly going to start. We have, I think, 30 seconds left to start in time. Please take your seats. You need a third one. Where is
Doug here.
Doug? There he is, yeah.
Where do you see him? Where do you see Doug?
Oh no, I thought he was coming in. Just yeah.
Perfect.
Yes, yeah. Okay, thank you so much for joining this afternoon session. It's really hard to get your attention. Okay, we're getting there. So please find your seats, especially here on the right-hand side from your side. There's still seats left. I already was in a panel this morning. My name is Andrea Meyer. I'm sharing this afternoon session. This afternoon, we have one panel again, 45 minutes, and then we have 3 short talks, so 15 minutes each. I think this panel we're starting with is very, very interesting because it's about the validation, and I think that's all what we need. So over to you, Brian Chen. Thank you so much for sharing this panel, and good luck.
Thank you. Pleasure to be here. Good to see old friends again. I'm Brian Chen, formerly with Luigi and Steve Horvath, and then ran off to industry. And now as of 2 months ago, I'm a free agent. Currently housed at UCSD. And to get here, we have Doug Keel from Harvard, Hebrew Senior Life. We have Aline Slagboom from Leiden. And we have Jessica Lasky-Sue from Harvard Channing, let's say it. And all of these are experts on biomarker development and validation. I think we spoke all together ahead of time. I think all the previous panelists before from this morning have raised a lot of great points, things that we would have raised. We're going to highlight some of those, and we're going to bring up some additional ideas. And then we're going to talk about, I think we want some more Q&A time, because I think a lot of people in the audience have some insights into validation here. So I'm going to have all the panelists introduce themselves, and then we have some slides that give a visual, since I know it's after lunch and everyone's tired, so we need to give them something exciting. So I think, and then each panelist